Ready?
Sure, yeah.
Welcome everybody to 52 Living Ideas. Tonight is Comprehensivist Wednesdays. This is done in conjunction with the Greater Philadelphia Thinking Society. Tonight we'll be covering, it's part of our neuroscience series, does the brain use statistics to operate? And as usual, Sanjay will be presenting to us this evening. Sanjay.
Hi, everyone. So tonight, what I'll do is I'm going to go into, this will be partly a review because there's a lot of information. We've been doing this for, it's close to 2 years now. Next month actually will be 2 years that we've been doing this for. And so I want to have a little bit of a review, some of the ideas, because not everybody has seen or been involved since the beginning. So some basic information that I think will be necessary, but that review is really for the specific topic that we're talking about tonight, which is a statistical type of information processing the brain does. And I'm not going to go into the statistics here. I want to make it clear that this is not really geared around math or traditional statistics. The word statistics here are used more in the form of patterns, because most of what the brain does is it processes patterns of information. So this, and the statistics will be in terms of the way information flows within the brain and how it disseminates, and there are consequences and repercussions of that we'll go into, and that I want everyone to understand. And one of the things that we do in this series is that this is an ongoing set of discussions we have, and the overall goal is to help everybody understand, several things. One is where the field of neuroscience is in a lot of its understanding. Also to give people a basic understanding of this, so that they, in our own ways that we operate in the world in terms of how we function. It's an adjunct to the knowledge of psychology that a lot of people have had. You may have taken courses in psychology, but it's a little more rare to take courses in neuroscience. So this is really meant to kind of add on to that, to help people understand a little bit, around why and how things happen in the brain, and that will help us to figure out why we do the things we do, why things happen certain ways. So one of the goals that I personally have, a lot of the people have in this group, is to look into issues like consciousness and free will and higher order functions of the human brain. But to do that, to understand that, we really have to have a better understanding of a lot of the dynamics that happen within the brain. A lot of the talks so far have been around trying to give a basic understanding of processes. And this is another one of those talks on, for example, the fact that information flows within the brain. And we've talked a lot about the fact that the brain is really a network of neurons. It's a network of between, on average 86 billion in adults to about 100 billion in children, infants. And with that many billions of neurons, there will be networks, and the structure of that network, etc., is really important. So what that means is that the information flows within the brain, flows in very, very complicated ways. It can sometimes loop back, circle back. It can go from region to region. It definitely does. So there's a lot of that, and I've talked about that in many of the previous talks. We'll go into a little bit of it tonight, but more so I want to go, this is more local in terms of just a small group of neurons, but the same ideas can be expanded across the entire brain regions and the entire brain. And another aspect that I don't mention here, that I won't be mentioning tonight, but it's evident, and we talked about this earlier, is that the importance of the brain is not only in terms of that one organ. The brain actually integrates with our entire body. And there are many aspects of the body that actually coincide with it, that integrate and together work. And every once in a while, I'll mention, even tonight, there's one or two things that I'll mention that shows how tightly coupled the brain and body are. So anyway, so let me start. What I usually do is I have a slide share. I don't think I have sound, but let me do that anyway. Okay, so I'm going to share the screen. Hopefully everybody can see it now. So the first thing I want to do is go into have an opening question, and the question is, how can information, and the topic we're talking about really, and we've talked about this many times, is that the brain really deals with information. That's the one thing that it works with. So how can information be stored in cellular tissue, right? And what that means is that information really it's intangible, right? We can't hold on to it, we can't touch it, feel it. It's invisible and it's non-physical. So in a sense, it's similar to what energy is. Another analogous question to this is, how can energy be stored in a battery? That's the same type of question that this is asking. So how can information be stored in a cellular tissue? So information is that cellular tissue is tangible. You can touch it, you can feel it, and it is physical. So how can something intangible be stored in tangible physical tissue? So this is a question I want to start off with. I'm going to pause the presentation. I want to get I want to give people a chance to answer this question. How can information be stored in tissue, in the brain tissue? So anybody who has an idea, if you've been in the chats before, if you've been in these talks before, you can type either an exclamation mark in the chat, or if you have your video on, you can raise your hand or physically raise your hand in the video, or you can use the raise hand feature in Zoom. Any of those methods, but most people put exclamation mark in the chat. That's the easiest form to manage. For Joe and for me, it would be easiest to manage. So if you have an answer, if you think and there's no right or wrong here, but I want to gauge the level of understanding that people have in this, because that will help me to, the level of detail that I should go into. So how can information be stored, information which is intangible, how can that be stored in tangible tissue? So I see Ron has his hands raised. And anyone else, you can raise your hand while he's talking.
So, since Nicole, you don't have an answer, then we're going to hold off on your comment for now. I have to give participants the ability to unmute. So give me a moment here.
Yeah, Nicole, we'll get to your question later. Hold on to that question.
Okay, so why don't we go with Ron, followed by Ella, followed by Myron.
Okay, well, the answer that just seems obvious is that it probably stores it similar to way our computers store information. They turn what's information in the same way you described it into something else. In the case of computers, I guess it's bits or whatever, but it obviously, the brain has to I would think the brain would do something similar to what a computer might do to store information. Am I on the right track?
So let me ask you what you mean, because for a computer basically has separate areas that are physically memory. And the purpose of that is for memory storage. But the brain doesn't necessarily have that. Every neuron in the brain technically can store memory. So is that what you mean, that every neuron acts as a memory cell in a computer? Is that what you're saying?
Yeah, I guess so, yes.
Okay.
So that's kind of you gave them the answer, right? Essentially, then with the neurons.
Well, no, I didn't give the answer yet. I asked him a question. So I want everyone, I'll give the answer at the end.
Okay.
And some of you may be right, some of you may be wrong. This is not about who's right or wrong. It's really about us understanding and having a better understanding of how it works.
But I guess just a little bit of addition to that question is, if it does have to transform that information into something else, and I guess neurons, it's mostly electrical current. So is it, I don't know much about the brain, that detail, but could you, if I'm on the right track, could you say what it might transform it into, that the brain could then use it and store it?
Yeah, so after everyone answers the question, then I'll go into it and I'll explain that. And then actually in the presentation, I have some slides that we'll go into. So if you don't have an understanding of how neurons work and communication information flows, we'll go into some of that tonight. So that's not a problem. Yeah, this is a learning tool and a discussion tool for all of us. That's why we do these talks.
So thank you, Ron. Next up, we have Ella, followed by Myron.
Hello, everyone. I was going to ask Sanjay if perhaps It could be on a cellular level, that there are possibly neurons in each cell. And I think I read a book that it was years ago by Bruce Lipton that was informative in that manner. I don't know if that, I'm sorry, I don't have more detail, but is it that each cell in the human body is sort of like in a primitive brain, perhaps?
Okay, that's an interesting thought. So I'll give the answer at the end. I don't want to comment on each person. So I want to get everyone's input. So I won't respond to each person right now. I'll respond at the end.
All right, thank you, Ella. Next up is Myron, followed by Ryan.
Hi, so I don't know, but I vaguely remember learning how the way neurons fire electrically is also a form of information storage. So in other words, maybe information storage isn't just a static process. The way the neurons activate can also hold information in a sense. That was it.
So we're gonna. You want to move on? Just take that answer in. Do you want to make any quick?
Yeah. These are all very good answers. Yeah.
Yes. So yeah, very good answer, Myron. Excellent. Next up, we have Ryan, followed by Judy. Be careful, Ryan, since you're on the road here.
Yeah, unmuted myself. See, the human brain is actually the least understood organ in the human body. And part of the problem is that you cannot dissect a person's brain when they're alive. It's only studied when you're dead. So that's part of the problem, but it's enormously complex. And most of the human brain is not understood. How information relays through its different parts and all of that, nobody really knows. Some things are known through studies, like, for example, gray matter versus white matter. And women have 10 times the white matter compared to men. All women do, 10 times. And a man has 7 times the gray matter as a female, 7 times. And that's huge. That's not some minor statistical variations here. Brain sizes are studied. Man's brain, a woman's brain weighs 85% of a man's brain on the average. It's smaller in size. You can study that, but you cannot come up with a real formula about how the brain tissue actually stores information and then how the messages are relayed within the brain. And that is missing.
Okay, thank you, Ryan. Thank you very much. Thank you for that. And next up, we have Vanessa, and then followed by Judy. And then actually, do you want to repeat the question for people that just come in, specifically Ian?
Yeah. So the question is, how can information be stored in cellular tissue? And specifically, why this is important is because information is intangible. It's not physical. And cellular tissue is tangible. It is physical. So, how can something that's intangible, something like energy, and the another analogy I gave is, how can energy be stored in a battery? So how can information be stored in cellular tissue? That's the question. So not everybody has to try to answer, but if you think you have an answer, this is where we're trying to get an understanding as a group on what the answer is. And after everyone gives their answer, I'll comment on everyone, and then I'll give the if we haven't gotten the answer yet, then I'll give the correct answer.
So we'll go with Vanessa, and then Judy, followed by Judy.
Well, I'm thinking it's possible that first, when there's maybe like a snapshot is kind of taken, you've got the electrical impulses going back and forth, and it could be a combination. Maybe it gets encoded, the intensity of the charge, maybe the frequency, and also I guess how many other connections like the synapses are formed in creating that. And the environment at the time may have an effect, like either the biochemistry, the temperature may strengthen the how that information is stored, like more of it makes an impression, so to speak. So I think it also has to do with intensity, the current and what the charge is at the time, the electrical charge and impulses.
Thank you, Vanessa. We have Judy followed by Jyoti.
Well, I don't know how it does it, but it seems like a miracle to me. So the thing is that what is it when we're talking about what is information? Are we talking about images, words, memories, feelings, knowledge? It's all this information and thoughts, So if somehow whatever it is, decides what to store and what not to store. So it's like a decision-making process. And that's how I see it. I don't know, I have no idea, but it seems like a decision-making process. That's how it does it.
Thank you, Judy. Jody.
Yeah, I'm just going to be guessing here. I think when the neuron activity is taking place, it converts the information into memory, and the memory filters down, flows down to the cellular areas. So that's where it gets stored.
Thank you, Jody.
Thank you.
So, Sanjay,
So
tell us the answer and who got it right and who got it wrong and who has to leave the meetup now and who has to. So
I didn't say anything.
Oh, Ian,
yeah.
did you want an answer? Do you want to answer, Ian, the question?
Attempt to. Am I audible?
Yes, you are.
Okay. This is a theory. We don't know for sure how information is stored in the brain. We have good theories with several evidence, but the problem is these theories basically seem to be falling short. Theories seem to have part of it, but it's more complex than them. That the special theories that information is stored in states corresponding to possibly binary, possibly higher, possibly mathematics higher base. Basically, the idea is that basically one state indicates one bit of information, another state, another bit that builds up like that, and these bits are encoded is thought in terms of possible synaptic transmission, firing across terminal axons of the dendrites along the synaptic gap. However, like I said, recent evidence indicates this may be the foundations, but it's far from the whole story. For instance, there have been a bunch of stories about computations within the dendrites themselves along protrusions called spines, which would enormously increase the complexity of the available states and the complexity of the overall mechanism, and the possibility of glial cells getting involved. Glial cells consisting of astrocytes, oligodendrocytes would make myelin, and microglia, which are thought to clean up debris and have a immune type function, were originally thought to particularly the astrocytes, which also play a big part in neurogenesis during embryogenesis, were thought to simply be supporting cells with neurons, enabling their functions. Now it's thought that they contribute also to the computational processes. So that's my answer in a nutshell. We seem to have the foundations, but it's far more elaborate than that.
Thank you very much for that answer, Ian. It's been it's a very, very good answer, detailed answer. Wonderful. So we also have Dwayne, is it?
Yes, sir, Dwayne. My background is computer science, so I approach everything as a math problem and as a technology problem. So it's the metaphor that I have to work with in my mind. And as I understand it, a group of neurons are interconnected to some level, and within themselves carry a signal bias for each of those connections, an individual signal bias for each of those connections. So a baby is born, either those signal biases in the brain of a baby are all set to the same default level, which would, if it was a computer program, that's the way I'd write it, or they're all set to some random level or some default genetic level, if such a thing exists. But over time, as you learn, as information is pumped through that network, then those biases change, and strong interconnections between neurons become more highly accepted, and less used connections between neurons become less accepted. And so the signal from one neuron to another changes over time with learning and with experience. So an individual memory, in my mind, would be ultimately the signal biases of the cells, the neurons involved when that idea. So if I look in the world and I see a car, then that visual information hits my visual cortex and creates a set of a network, an array of input signals that touch a bunch of neurons in my head, and the signal biases taken together return with the value car. And so that's how I recognize things. So babies are born not recognizing anything, and over time, people say, that's a car. And so that signal set becomes car in their mind. This other signal set becomes mama and food and whatever else. So ultimately, memories, knowledge, everything in the brain is a set of bias values among neurons, I would think.
Thank you very much for that answer. So, Sanjay.
Yeah, so very good answers, interesting.
Very, very good
So
answer.
I'm going to give a simpler version. So two people I heard answers that were much closer to how it happens. Duane, who just answered right now, and Ian before that, they both gave answers that roughly correspond to what's happening. And I'll succinctly explain it so that everybody else can really understand what they said. Although there is a little bit of a variation. What they said isn't 100% true, but I'll explain that aspect. So basically, when we're talking about information being stored in the brain, let me first explain that. I think it was Julie who talked about the different types of, whether images and words and memories and thoughts, these are what information is. And so in each of these talks, I've explained a lot of things in prior talks, so it's not possible in 2 hours even to go through all of the types of things that I've described and other people have described or learned in these talks. So just succinctly, I'll say that information is anything that goes into our brain from our sensory input. So from our, for example, balance. Our body needs to balance. When we're walking and standing, our body balances. So we have sensory organs in our ears, the cochlear organs, which give us a sense of balance. That's information that comes into our body. Our eyes, ears, nose, mouth, etc. obviously have information that goes into our brain. And then there's also information that is already in our brain from before, which in the form of memory usually. And then there's also information which is from our body, from our gut. So for example, if we're feeling hungry, there's a deep connection between our body and our brain. So information comes from any of these sources, and all of these are information. And information can be either the most simplest. For example, the sensation that we're hungry is the simplest form, one of the simplest forms of information we can have. Basically, it's either on or off, right? Either you're hungry or you're not hungry. And for some people, it might be a level of hunger, although technically it's not, because what it actually is, it says the frequency, how often it sends it to your brain, is how we interpret it as a level of hunger. It's basically it's sending a signal or not. And if it's sending it every 2 minutes, then we feel very hungry. If it sends, once every 15 minutes, then we're not as hungry. It's that type. But basically, that's one of the most simplest types of feelings that we have. Other things, for example, visual input, odors, complex memories, dreams, occurrences that happened in our life. Those are all much more complicated information. So information, all of these things are information. And the way it's stored, so just there's some misnomers that I heard from several people. Something that Ryan said, actually, we do understand the brain a lot more, and there's a lot of understanding. I'm not going to go into it if you don't understand that, or if you want a better understanding of it, you can either go to the talks that we've had at this group, or there's plenty of sources around the internet and research papers that you can use. But basically, we understand. Now, this is not to say that we understand everything about the brain. We understand, compared to everything that can be understood, we understand very little of it. So compared to the enormous amount of information that's possible to understand, we might understand less than 5% of it, less than probably 1% of it. But even the level that we understand actually helps us to have it gives us predictive value. We can actually predict a lot of things about patterns of behavior in people, and we understand why the brain does certain things. So in terms of, another piece of information I need to provide before that is that I'm trying to clarify some things that people said. So the brain consists of many types of cells. Okay, and our body basically is composed of cells, all very different types of cells. And our body has multiple types of cells. One type of cell that's in the brain is called a neuron. A neuron is simply a cell, a single cell, and it's a very specific type of cell. Our brain has other types of cells also, and Ian referred to those. They're astrocytes. And there are many types of other. They're support cells, which don't actually. They're not involved in the processing of information. They're support cells that help our brain to stay alive. And there are, for example, immune cells. They're actually a separate immune system in our brain, separate from our body's immune system, believe it or not. And so there are immune function cells in our brain also. So there are many other types of cells in our brain, but the main type of cells that we're talking about, that we're interested in, are neurons. And neurons are specific cells which carry information. And there are actually several types of neurons also. There isn't only one type of neuron. And the shape of the neuron, specifically the types of axons that it has. Now, I have some slides later on that will give a cartoon image of it to help explain this, but roughly all of these neurons are connected together. That doesn't mean that every neuron is connected to every other neuron, or even that one neuron is connected to all of its neighbors. That's not what it means. It means that any given neuron is connected to several, thousand of its neighboring neurons, because it has tens of thousands of neighbors. So a single neuron has tens of thousands of neighbors, and it's connected to probably around a few thousand of its neighbors. So it's connected to a small subset of its neighbors. And if you pass that on every neuron to every neuron to every neuron, the entire brain is this huge connection. It's called a connectome. That's the word that's used in the neuroscience. It's basically the map of how every neuron connects to every other neuron. It's a huge map, and we don't have that map. And that map is not fixed for every person. It varies from person to person. But there are rough patterns from people to people that it follows. So if you understand so far that we have this immense connection of neurons, okay, then the answer is that all of the information that's stored, it's stored between the neurons in those connecting paths. That's the important thing that I want everyone to understand. And that's a vital piece of information, because in all of the talks that we have, and if you go back and look at prior talks, you'll need this information, and in future talks, you'll need this also. But that's the most important aspect that you have to understand for us to go forward in any area of neuroscience, is that information is stored between the neurons, it's not stored within a neuron. Now, there are aspects that Ian hinted at some of this. There are aspects, and actually I think Duane also, there are aspects that specific neurons can impart in the way that information is stored and in the way that information is passed from neuron to neuron. But that's not really technically where the information is stored. Those are more around biases of individual neurons. And actually, there's a lot more complexity behind this. There are neurons that actually work reverse. There are excitatory neurons and there are inhibitory neurons. We've talked about this in the past, but I'll bring it up tonight because it looks like there are people that aren't familiar with these. The complexity of the brain is enormous. And I'm trying to simplify it to the level where people who don't have a background in this can understand it and it can be useful to them. So the key idea again is that the information. So for example, the memory of a car. I forget who, somebody gave an example of a car, right? And a baby, is not born with a memory of a car, and a baby probably won't form a memory of a car in its first few years unless it sees the same car repetitively. But even then, it won't recognize it as a car. It'll recognize it as almost like a photo, the same way it recognizes the photo of a star. It doesn't know what a star is. It doesn't know what a potato chip is. because technically babies shouldn't be eating potato chips, right? So there are many things that it sees, but it doesn't understand what they are. It simply sees them as a static image or as a moving image. So it doesn't have a concept of a car, but it might recognize the shape of the family car and the color of it and the fact that it sometimes makes a sound. It might recognize those aspects. And that information about that car, as the baby grows, over months and initial years, there will be many, many neurons, millions of neurons that together will encode various aspects of what it is that their family's car, that baby's family's car has. And that information will be stored between those millions of neurons, not in any one of those neurons. So that's important. So let's continue. So any questions so far? Let's just do a quick Q&A just to make sure there's no confusion on what I just presented.
So folks, go ahead and type exclamation point in the chat or raise your hand in Zoom if you have any questions specific to what Sanjay just said. Please hold off on your comments for now, just any specific clarification questions at this point. I have to give people the ability to unmute themselves. So we have Ryan followed by Ron, and then so please be brief. Both Ryan and Ron.
I guess my question is, how do you explain people that have intuition? So there's no background, there's no knowledge, something new they discover, they've never seen it, completely abstract as a matter of fact. And then they're able to come up with, like in mathematics, they do things out of nowhere. They pull them out of a hat.
You're muting and unmute. Unfortunately, somehow I'll ask you to unmute really quickly. And
yeah, your audio is cutting in and out. So let me just say, that question.
So
that question isn't it? Sorry, Joe, go ahead.
Oh no.
Can you hear me?
Now we can hear you. So your question is specifically about intuition, right?
I'm going both ways. So one is the mathematical genius, and the other one is saying that a man and a woman is a social construct. So obviously you can look at a man and you can look at a woman, and the neurons have stored that information. They're telling you, but they're saying, no, that's just a social construct. So how do you get to that point, right? These are two extremes on the opposite ends.
Okay. Thank you, Ryan.
Sanjay, so Ryan, or everybody in general, right now I want to focus just on what we talked about so far. Ryan, we'll get to your question. I want people to ask questions, but we have to remember that question like that will have a very complicated answer. And if you don't understand some of the basics around brain physiology, brain function, etc., then it won't make sense, or you'll have an incorrect understanding. So that's why it's important to, these are learning sessions. These are, opportunities to understand what's understood about the brain. And, sometimes intuition is much more complicated. I'll go into it later, but I don't want to go into, any and every type of question that people might have right now. I want to focus just on how information is stored between the neurons, that aspect. If anyone doesn't understand that or has a question, that's what I'm asking people to if you want clarification.
Yeah, and I'm going to ask everyone to please be brief here because we have 4 hands up and we still haven't gotten to the presentation. So I really want to, it's 9:40. Actually, Sanjay, do you want to continue with that? Because I think this could turn into, it won't be 10 o'clock until you start. So why don't we hold off? Do you want to hold off until the 25 minutes from now? We can do it during a Q&A then.
Let's give everyone about 10, 15 seconds to ask a question.
Okay.
Very quickly. Just one sentence for your question. Yeah, if you can.
Okay.
And if it's not answered, then during the talk we'll have other opportunities for questions.
Okay, everyone, you'll have opportunities for follow-up, but for now, just a brief 10-second question, if possible. Go ahead, Ron, followed by Dewan.
Okay, Sanjay, so you said that the neurons encode information. So if we see a car, how does a car get, quote, encoded into a neuron? What the heck does that mean?
Okay, so if I could say, information is not encoded in the neuron. Nothing is encoded inside neurons. They're encoded between neurons. Mm-hmm. That's the key.
Well, okay, it's the same question applies, doesn't it? What is the encoding process?
So we'll go into that in the talk. We'll go into that.
Okay.
But I'm not going to go into extreme detail because this is not a course in neuroscience, although we can talk about it. But for you to understand really how that functions, you have to remember that any answer that I give you will raise more questions, right? Which is great because that's why I want people to think about this. But this is not an opportunity for you to learn all of neuroscience. We can't do that in 1 or 2 hours, right? Hold these questions, and in future talks we'll elaborate on these questions and ideas that people have. Vanessa?
And I think actually I think I lowered his hand by mistake. Did I lower
Okay.
your hand by mistake?
No,
there?
no, I did. I did
Oh,
my,
okay.
I don't actually have a question about this, so I
Okay.
just wanted to bow out. And it's
All right.
Dwayne.
Or Dwayne, I'm sorry.
That's all right.
Forgive
I'm not
me.
making-
Yeah, forgive me. Vanessa, followed by Mike.
Is this kind of just like the basic information, the initial impression of it, or does it also include like information that's maybe being combined with other information, make something more complex? Like, is there a difference between, or it doesn't depend on the type and complexity of the information?
So again, that's a different question. I can answer it very quickly. That a single neuron the single neuron has multiple connections, and each of those connections can store different types of information. So if a neuron is connected to 1,000 other cells, technically it's holding, you might say, 1,000 pieces of information. It's not that simple, but it's holding many pieces of information. So that, in a sense, answers what you're asking. That the multiplicity of information that our brain stores. We'll get into that a little bit later, but that's a rough answer. Mike, just very simple. One in 15 seconds, if you can ask. Mike, you're muted.
Yeah, when we first started this, I sort of thought that you considered that there was one bit of information stored in a neuron, and I challenged that. Now I see us evolving to the fact microtubules between and synaptic junctions with millions of pieces of information recorded in a single synaptic junction. Is that what Macram and Penrose are moving toward? Thank you.
Yeah. So again, you're getting into much more complicated areas that many people might not grasp, but we'll go into that later. I don't want to go into that tonight because microtubules and exactly the details of, the way information. Basically, what Mike said is that a single synaptic junction, which means a connection between any two specific neurons, the connection between them, there's a point at which they quote unquote connect. They never touch, but they quote unquote connect. And that junction, that area, actually can encode more than one type of information, but it's not infinite. It's not a very large number. And it can actually encode information in reverse also. It's very complicated, but we won't go into that. But basically, this is the reason why the brain is so powerful, because it can encode more than one bit of information or more than one type of information within the connections that emanate from a single neuron. Let me hold off here. I hear a lot of the questions are on peripheral or other topics. Let me go into the talk itself, because that's if we don't go into the talk itself, we're not going to have questions and we won't have an understanding. So I want to make sure that people get an understanding of what we're doing. So
Okay.
let me.
What do you want to hold off on Ian and
Ram? Yeah.
Okay.
Yeah. I think
Ian and Ram, if you could patiently wait to ask your questions. We're going to have a Q&A period in about 25 minutes roughly, so that you can ask Sanjay or follow up with anything he has to say at that particular moment in time. In the interest of time,
also feel free to type your question in the chat. That way it can be read and when you get a chance, you'll be able to.
So apologies that you didn't get to answer or ask your questions, but we're trying to keep to a schedule here. Do you want to start sharing your presentation again? There was a little bit of a issue with the way the presentation was actually coming up. I don't know if that was on your end. It was just a little bit of yes, there it is.
So is it fine? Does it look?
No, it's got a black box that's covering like the opening question. Do you not see that?
I don't see. My screen shows everything. I mean, Zoom has the overlay, which shouldn't appear on.
There we go. Well, but it's happening at the bottom now. It's odd.
So you know what? That's the Zoom overlay. That's actually
Okay.
what's, you know, I don't know if when you share a screen, there's an overlay that appears on my screen that lets me control things.
Yeah, there it is.
That's
again.
the box. I just moved that box below. So I don't know why that's, so I'm going to move it to the middle. So is it in the middle now?
It is. It is.
Okay, so that's the box. I don't know. I can't move it off screen, and I don't know if I can get rid of that box. I'll try to move it.
Can you optimize? If you optimize it, click the optimize button for sharing. Does that do anything?
I don't see any optimize.
It should be right next to the sound when you go to share screen.
Under video settings?
Yeah, it's under the share screen settings.
Yeah, there's no.
There should be two little boxes. One's audio.
No. That's before I started the share. Those two.
Okay.
Yeah, so I already did that. I optimized
All right.
the video.
Okay.
I did both of those.
All right. That's what I
But
was
during sharing, it puts up a panel of many buttons that help me to control. For example, if I wanted to annotate on here, that's that. You know, it's a long rectangular box. That's the one that probably is appearing. I don't know why. I don't think Zoom has a different version. I mean, I'm up to date. Anyway,
it will be problematic if it's that big as the previous slide, but why don't we see what the next slide? Yeah, now it's completely covered.
It's what's completely covered? The whole screen?
The whole screen was covered for a moment there.
Okay, so
that's workable, but if you have a graphic up there that's trying to present.
So is it more than 5% of the screen? Like
it's a
horizontally?
It's around 5% of the screen. It's maybe a little bit more than that. But.
So what I'll do is I'll move it around. If there's anything at the bottom I need to show, I'll move that away so people can see it.
Okay.
But most of the information shouldn't be there, I think.
Okay.
So all right. So basically, let's first go into the fact that the brain processes and uses information. Well, brain processes use information. So processes, major processes that we have in the brain are things like sensation, perception, and experience. These are things that we've talked about in past talks. Memory, memory where you're storing information that you don't know or haven't experienced yet. You're remembering things from before that you've already experienced. Confusion, delirium also are aspects of memory. Memory is actually a complicated area. Cognitive activities, cognitive processes, for example, understanding, the fact that you're understanding the speech that I'm saying, because we all speak English here. You may be reacting, within your mind. You're not reacting in your body, but within your mind, you're having thoughts and you're having understanding. That's a reaction. So that happens in our brain. Decision making. I'm making a decision on when I should click the slide, when I should do things, what I should say. So decision making. These are all processes, examples of processes that happen within the brain. And then muscular activation. When we move any of our muscles, right, including when we talk, when our eyes blink. Even the fact that our heart beats, although heartbeat is a little more complicated because there's autonomic activity there also, but our brain basically controls a lot of the muscles within our body, most of them, to actually cause, and oftentimes not just muscles, other types of things also, to cause our body to do certain things. So those are all major processes that are part of the brain. And then there are also areas of the core brain, which is known as the brainstem. There are many functions, for example, respiration, certain various types of regulation that the brain basically regulates and keeps the body alive and intact and stable, etc. Sense of balance I talked about earlier, the sleep wake, whether you're conscious or not. These are aspects of brainstem. And there's information that flows from the brainstem to the body and back and forth. So there's a lot of information that's flowing based on a lot of these processes in the brain. So Joe, is anything covered yet? Or is that below the?
You're
okay,
good. You're good right now.
So
the core brainstem should be the last bullet, right?
Yeah. Yeah. So that's kind of where most of the slides, sometimes with the video, it might go a little lower, but most of the video is in the center of the screen. So the next is information flow, right? So how does information flow in the brain? So information flows within the brain. That's important to understand, right? So one of the things that happens with information is that you have to have awareness that information is flowing in the brain. You have to have awareness, for example, that you're listening to me, right? If you're asleep, then you won't have awareness, even though, believe it or not, even when we're asleep, information is still going into our brain, and it's causing, flow information, that information, that sound that we're feeling may go inside our brain, but it may not be awareness, okay? Another example is if you're sleeping and somebody touches your body, right? Well, if they touch it just lightly, you might not be aware of it. But if they touch repetitively or they touch it with a certain amount of force, all of a sudden your brain, a part of your brain becomes aware of it. Not that the entire brain wakes up, but a part of your brain becomes aware of it. So the level of awareness and the fact that we can be aware or non-aware, this is very important. And this affects whether and how information flows within the brain. The emotions that we have. So emotions are another type of information, whether you're happy, because when you're happy, it comes out in our face, right? It may come out in our eyes, or our mouth may smile. The way we walk, our gait may change. You have a step in your walk if you're happy. If you're not happy, if you're sad, then your body may hunch. So emotions flow within the brain, and then they flow into the body also. So emotions are types of information which flow within the brain. Thoughts, we've talked about thoughts. The fact that you're listening to me, and the sounds, the phonemes that your ears are picking up that are going into your, the temporal lobe, they're being processed. They're being converted into specific words and sentences which have meaning. And those meanings are basically a thought. So you are having thoughts based on what I'm saying. You're having an understanding based on what I'm saying. So these are all elements of that. And then we have memories. Many types of memories. Some examples here: procedural memories are, how do you drive a car? How do you eat with a fork? Right? These are procedural memories. Spatial memories are where your house is relative to your office, right? Or where one room is relative to another room in your house. You know that. You don't have to learn that every time you walk through your house. You already know that. That spatial information is encoded within your brain, in your memories. Episodic memories: what happened when you were 15 years old? What happened yesterday? These are episodic memories, episodes of occurrences of things that happened. So each of these, the information flows primarily from certain areas of the brain, but they flow throughout the brain. So awareness is throughout the brain, but brainstem is one of the most important areas where awareness is central for information flow. Emotions tend to be mostly toward the subcortical regions below the cortex, the amygdala, the many regions, hippocampus. There are many regions underneath the cortex. Thoughts are primarily cortex, and memories are cortex and hippocampal mainly. But this is a very rough picture of how information flows and where they flow primarily from. Although each and every one of these types of information do flow and can flow pretty much anywhere and everywhere in the brain, almost anywhere and everywhere. The subcortical regions are a little more limited because they have very specialized functions. And one of the talks that I did over a year ago actually talked about the brain. Many people don't realize we actually have multiple brains. We don't have just one brain, if you think about it. Or one way to think about it is that we have multiple brains. And these brains evolved over time. So when we were very early mammals, our cortex was not very developed. We hardly had a cortex, and some early mammals don't even have a cortex. So most of their brain function was at the subcortical regions. That was the primary brain. But now we have that subcortical region, which we've retained through evolution. We still have it, and it still functions the same way it does in a rat, let's say. Okay, but we also have this thing called a cortex, which is a thinking brain. So at the simplest level, we have two brains: our thinking brain and our emotional brain. So that's an aspect that's important in this. What we're saying here is when information flows, information simultaneously flows throughout many different regions and many different levels of the brain. That's what's important to understand here. Next, this is a video that I think I may have to start it. This is a video that shows basically how this is actual living brain and how information. Actually, I thought. So this is showing, it is an animation of a normal human brain at rest. There's not much going on. So even when our brain is resting, when it's not really doing anything, information is flowing throughout the brain all over the place. And the colors correspond. The blues are basically where there's very little information signal flow, and the yellows are medium level, and the reds are where there's a lot of information flow. And as you can see, the information is flowing in repetitive patterns almost. And this video is basically a loop that's repeating. So in reality, it won't be an exact repetition. It will be, roughly speaking, many different areas will be activated and deactivated and partially and fully, etc. So this gives a representation of what actually happens within every person's brain at rest. This is not when you're doing something specific. So during a speech, it would be much more different than this. This is basically, you might be, daydreaming, or it might be just just laying down and just looking at the clouds, pass by overhead or something. It would be that type of activity where you're not even trying to think about things. You're in a passive state. And even in those situations, our brain is always, information is always flowing from area to area. And one of the things that's important to understand is that if our eyes are open, information is always flowing to our occipital region. And in this model, the left side is the occipital region where a lot of visual activity happens. And so if we're seeing, there's always information in the rear of our brain, the occipital region. And that will be causing us to notice, what we're seeing in front of us. If something is moving in front of us, that motion will cause certain activities in the brain also. So vision is a very complicated set of processes. It actually takes up almost a third of our brain. It's a very complicated part of our brain. So there's always activity. So now let's go into a little more of what actually, is a neuron. So this is a very simple cartoon diagram of a single neuron. And for those of you who took biology, our body basically is made up of hundreds of billions of cells. And a neuron is a single cell, except this cell will look different than the type of cells that you saw in biology class. In biology class, you may have seen something that basically was kind of circular, or like an ovoid or something. It's basically like a football shaped, or even a soccer ball shape, or tennis ball shape. It's a round thing in a two-dimensional flat image. It'll be a round thing, and inside there'll be a nucleus. So a neuron has that aspect of a cell. It's basically, this is a soma. This is the main part of the cell of a neuron. And then neuron also have two other parts which are very important. They have this thing that's sticks out. It's a long projection, and this is called an axon, AXON. And then it also has these things on the outside which are called dendrites. And this is a very simple cartoonish image of it. Actually, and I have an image of an actual neuron later that will explain it. But this is a very simplified version. So all these dendrites basically come off of the soma, come off of the main cell. And there's one axon that comes off of the main cell. And this axon can be very, very long. Now, believe it or not, this axon can be, in most humans, we have one or two axons which are about 3 feet long. Okay, meaning this axon itself is 3 feet long, and it goes from our brain to our toes, and it goes through our spinal cord. And in some people, it's actually split into two, so it's 3 feet. And it's very rarely, it's actually a single cell which goes the entire length, 5 to 6 feet long. So these axons can be immensely large, although most of them are very, very small. Most of them, these are on the order of a millimeter, or a few millimeters. Although somebody asked about the corpus callosum. The corpus callosum is basically a set of interconnections between the left and right hemisphere of the brain. Our brain has two halves called hemispheres. And the corpus callosum is a region kind of in the middle, in the central part, which has all of these axons, these nerve fibers. And these axons basically is what we call a nerve. Although nerve is a bundle of hundreds of axons. Hundreds of these coupled together into a single long fiber would be what a nerve is when we think of a nerve. And so the corpus callosum that connects the left half of our brain with the right, and vice versa, interconnects both halves. There are many of these nerves that go left to right, right to left, between both halves, and they basically are only or mostly these axons and clustered within nerve bundles. And so those tend to be more than millimeter, obviously, because the brain is about 8 inches wide. So it can be from several inches long within the corpus callosum, but within most regions of the brain, they tend to be very short, within a few millimeters at most. So next, the information that flows within a neuron is very specific. Okay, this arrow shows information always flows from the dendrites into the cell body, through the axons, and then out to the end of the axons. And these things are called the terminus or terminals. These are where they end, the axons end, and the information goes to these ends. Okay, this is very important to understand. Information only flows in one direction in every neuron. It can never flow the other direction. It's impossible. Biologically, it's impossible.
Right.
So this is very important to understand. Joe, is there any question? I can't see participants.
No, I accidentally commented, so I apologize.
Oh, no, that's okay. No, it's good that you emphasize, because I want people to understand the importance of some of these ideas, these initial ideas. And again, I'm doing this as a review. I've talked about these prior to this so some of you, I'm sorry if this is new, but I think this will be a refresher for everyone, because these are very important things that everyone needs to understand. So information always enters a neuron through one of these dendrites. So information will enter, and you'll see that this is almost like a hare's nest. It actually, this is known as a dendritic tree. So this dendrite actually is more on the analogy of a tree. If you think of the branches of a tree, real dendrites are more along the lines of, if you think of a tree here, and all of the leaves and the branches coming out. This is a very simple image, so it looks circular, but it's actually not circular. It's actually more shaped like a tree, and the tree branches spread out all over, in directions. But generally, all around the cell body. So information comes in to a neuron, and it has to come in from a dendrite. Okay. There are some exceptions where it can come in other ways, but I won't go into that. But typically, most information that enters a neuron enters from the dendrite. That's what's important. And then the information flows through the dendrite. It eventually goes to the cell body. And then if the information is strong enough, and I'll talk about that in a minute, if it's strong enough, then that information gets passed through to the axon. If it's not strong enough, then nothing happens. The information basically goes into the neuron, and in effect dies. It not dies, but it dissipates. The information gets lost. So neurons don't really hold information. That's something important to recognize. They don't hold any information. When the information flows through them, they pass through the cell body, and if the information is strong enough, then it immediately passes, immediately meaning within a few milliseconds, they pass through the axon, and then they pass to the terminus. And these terminus are connected to another neuron, and the next slide will show that. There's going to be another neuron with its own set of dendrites over here. So that's where, if somebody has a question about where the information comes in initially into the first neuron, they come in from dendrites that are at other neurons. And then you might ask the question, well, what if there's no other neuron here? Well, there's always something here. And the other type of thing that we might have, if it's not a normal neuron, it could be sensory cells, meaning, for example, our taste buds. Our taste buds are gustatory. They're similar to a neuron. They're a little bit modified in that the information that they collect is molecular. They reside mostly on our tongue, and they collect information. They turn that information into information. Or sorry, they turn the taste sensation into a signal, into electrical signal, which gets passed through it. And then they also have these terminuses on the end, which will eventually reach, into a single or a small number of neurons. And then our taste bud will, that information from the taste bud, the sense of whether it's a sweet or sour taste, that single piece of information will go into a specific neuron, and it'll get passed on into the network, into this vast, huge network of the brain.
These are chemical signals, Sanjay?
So I'm going to go into that in a minute. Yeah. So
okay.
The way that the signal is transferred, as soon as in the next slide, I'm going to go into, I'll have multiple neurons, and I'm going to go into how they pass from neuron to neuron.
Oh, great.
So yes, it is chemical, but I'll describe that a little more. Now, we're not going to go to extreme details, but we're going to go into enough detail that you understand the complexity of this. And also, one of the things that I want to point out is that in several of the talks that I've done, I try to build parallels with neural networks, which are a form of computing. So Dwayne talked about computers, and there may be other people in the attendees who have a background in computers. Neural networks are different from traditional von Neumann-style computers. They are neural-based. They were network-based computing. It's a very different model, and the model is based on how biological neuronal tissue functions. But neural networks that we have today are very, very simplified versions of it. yet they still perform very, very adequately. They're making immense headways. And I every once in a while I'll do a talk which ties parallels, because some of the ideas in computer science area of neural networks actually apply to brain physiology and brain function, and vice versa. So I use both. So some of the talks will span a broader area, a broader range of ideas. Information in, for example, the computing neural networks are very similar. They have basically, they don't have dendrites necessarily, but the neurons are interconnected in a similar way. Now, what's important about the dendrites is that information doesn't have to come in only from one dendrite. You can have multiple pieces of information coming in simultaneously into the same neuron. So here I show 3 different arrows. You can have 3 different other neurons, right? Each one associated with this path of flow. So if there are 3 other neurons to the left of this one neuron, each of those might fire simultaneously, and there might be 3 pieces of information that go into this neuron. And if that happens, then because the amount of information flowing is higher, this neuron is probably more likely for that information to pass through and get passed on to its axon and into other neurons that it's connected to. So this is the first point at which we start to see statistics come into neuronal function. So statistically, there's a sense that if all 3 of these input neurons are firing simultaneously, then there's a certain chance. I don't want to go into the mathematics or the probability of it, but there's a certain increased chance, because you have multiple things happening. There's a greater chance that this neuron will be active or activated. Now, if these 3 neurons don't fire simultaneously, but they fire in succession, one, and then it turns off, and then second, and then it turns off, and third, it turns off, it might give a complex nature inside this neuron, inside the cell body. And depending on the intensity of the first signal and how long that first signal lasted, and the intensity of the second signal and how long it lasted, the intensity of the third signal and how long it lasted, and the delay in time between each of these successive signals, right? So now you see this is getting really complicated. If you had 1,000 different interconnections with this one neuron, as I described earlier, a single neuron can have thousands of other neurons that tie into it. So if you have thousands of these, this becomes a very complicated statistical process of when this neuron actually fires. And that's at the simplest level where statistics comes into our brain function. So the next level is that, well, let's say that for whatever reason, there's enough information that flowed into this neuron, and then that information gets passed on. Okay. Now the information that flows into this axon we can think of it as electrical, but it's not really electrical in the true sense. It is not a current. Okay. It is a voltage, but the voltage is different than current-based traditional V equals IR, the traditional electrical type of signals that we have in computers and electronics. This is ionic. These information is carried through ions. And somebody earlier had mentioned microtubules. Microtubules are what exist, although they exist in many places, but the most important area in brain, microtubules exist throughout the axon. And they're a very important part of the axon, and that are actually what help to carry this information through the axon and in one direction, because microtubules, the way they function, they behave in one direction typically. That's the reason why the information flows primarily in a single direction. The information actually flows with inside. So this axon, you can think of it, actually consists of many hollow things inside it, hollow channels, you could say. And one of those hollow channels is called a microtubule. Although there are many types of microtubules, and each axon has hundreds of microtubules. So many types of information can flow within this. But typically, the information that flows, I'm not going to go into detail because this is going to. I don't want this to be too complicated for people for whom this is new. But another idea that I will present, which is important, and this is this is throughout the neurosciences, is the idea of neurotransmitters. I'm sure some of you have heard of neurotransmitters. We have many types of around 30 neurotransmitters that are common in the brain. Many of them are very, very prevalent. Dopamine is one, oxytocin, norepinephrine. These are common neurotransmitters that exist in the brain, in the human brain. And they are actually, a neurotransmitter is basically a molecule. It's a small molecule. There actually are other types of molecules also, which can be neuropeptides, or in general, they're called neurochemicals or neuroactive chemicals. There are many types of molecules that are used to carry these signals, But the key information is that each of these molecules flow inside these axons, as well as there are ionic signals, for example, potassium and calcium molecules. So one of the reasons why we have to eat salt in our diet, one of the reasons why we have to have potassium and iodine, other types of minerals and nutrients in our food, in the food that we eat, is because they're important for brain function. They're important for signal transduction, for the movement of signals from neuron to neuron. These are very important molecules. So if you don't eat enough salt, this is the reason why it affects our heart, because the signal that goes from the brain to the heart to regulate the heart pulse and timing is a calcium signal. And if you don't eat enough salt, or if you eat too much salt, the amount of salt and calcium in your body can be either out of balance or imbalanced, and that can disrupt heart function as well as many other types of neuronal functions. So that's one of the reasons why if your physician tells you to change the amount of salt intake you have, increase or decrease, because it's one of the very important chemicals, molecules for brain function, for nerve function. So many types of signals actually flow along the axon. I mentioned neurotransmitters. I mentioned ionic signals in the form of either calcium ions or potassium ions, or several types of ions that are important here. Now, another thing to understand is that every axon doesn't have every one of these types. The types of neurotransmitters actually flow within a specific neuron, depends on the type of neuron it is, and it depends on many other aspects, which we're not going to go into tonight, but it depends. It's very complicated, and again, that's another layer of statistics, in that if a specific neuron is a certain type, and there happen to be a lot of those neurotransmitters that it needs, because the neuron basically gets neurotransmitters from outside. Sometimes it can manufacture the neurotransmitters itself, but typically it's absorbing the neurotransmitters from outside. And let me explain this, that the way it absorbs the neurotransmitters is what these blue arrows show. These blue arrows are actually neurotransmitters flowing. That's how the information flows from a neuron that's before this. That neuron at its axon terminus, it'll send out neurotransmitters. And I mentioned 30, there are 30 various. Typically, a single neuron might have 2 or 3 or 4 types of neurotransmitters that it deals with at a time. And a different neuron might use a different set of 3 or 4 neurotransmitters or neuropeptides. And so, depending on the interconnection of neurons, the types of neurotransmitters that are prevalent all around this one neuron, this neuron is basically swimming in a sea of neurotransmitters, depending on the other types of neurons that are around it. Now, if all the other neurons are very similar to this, then you probably won't have all 30 types of neurotransmitters. You'll probably have maybe 5 or 10 different types of neurotransmitters, because this is a localized region of the brain. But if you look throughout the brain, all across the brain, the brain has all of those types of neurotransmitters all throughout. So next slide, so this is a little bit more detail where we have 3 different neurons. If you recognize, again, we have the cell body with the dendrites around it. We have this long axon, and then we have the terminus at the end of it. And then the terminus comes close to the next neuron, which has a cell body, and then it has its dendrites, it has its axon and the terminus, and then that comes close to the third neuron, etc. So in this case, what you'll see is that the information flows from one neuron in one direction. Remember, it's always in one direction, into the next neuron, and again, it flows in the same one direction, and then into the third neuron. So information always flows in one direction throughout the brain. This is very important, okay? And it always flows in a specific way. Information can never flow backwards. Okay? There are ways that the network can be built. So for example, you can have a neuron that's backwards here. Let's say that we have a neuron that's backwards here. The head of the neuron is here and the tail is over here. Well, then when this neuron fires and the signal goes to its terminus, it activates that next neuron here, the head. That might fire and then activate this one again. So you end up with this cycle. Imagine the second neuron here. It'll activate itself over and over and over. Technically, that can happen. But in that case, the cycle will be that it will still be flowing in one direction. It will be a counterclockwise direction in the example that we have here. So what's important to understand here is that the neurons aren't alone by themselves. Neurons typically have many other connections. So I'm just looking at this first neuron. This first neuron might have 2 neurons on the end of it. It might actually have a third neuron on the end of it. And the direction of these neurons can be in any direction. They don't have to be necessarily always going in one direction. They can flow backwards also. So the fact that the signal comes out of this neuron and goes here, it can go into this neuron. It can also go into this neuron. And it can also go into this neuron, because remember that what comes out of these terminals are neurochemicals, right? They come out and they float around this neurochemical. Okay, and the first neuron, the second neuron never touch. That's very important. They never touch. Okay, if they do touch physically for accidentally, what happens is one of the ends actually dies. It atrophies because they're not supposed to touch. So they actually and this is very important, that the neurotransmitters spread. It's almost like a cloud. If you think of smoke, or if you think of perfume, right? The molecules of perfume come out and they pervade the area around this. Okay, so if you think about this now, if you understand what's happening so far, let's say that this set of terminals actually have lots of neurotransmitters emitting here. And let's say there's a immense profusion of chemicals that are transmitted here. What's going to happen is that these neurotransmitters are going to cluster around. It's going to grow and grow. Think of somebody having a very, very strong perfume. The scent or the odor, expands and it spreads out over space. And the same thing happens here. It spreads out over space. So if there's enough neurotransmitters from this, now this terminus might have an intention to only communicate with this one neuron. But if the signal is so very strong, or if the signal is so very repetitive that it keeps neurotransmitters from being activated over and over and over and over, that there's such a strong amount of neurotransmitters that are built up in this area, and those neurotransmitters will not only pervade into this one neuron, which it's intending to activate, it might actually also activate the second neuron, and maybe even this third neuron, which is physically more distant. So this is what's important to understand. This is another level of the statistics or the patterns. So the fact that these neurons are different distance from each other, and that their positioning and their angle and the direction and the level of interconnection, for example, these two neurons here are not connected very strongly because there's physically a large distance between them. These two neurons are very closely connected because you can hardly see any gap between them. There is a gap, but you can hardly see it because they're very, very tightly coupled. Although this neuron here is not coupled with the other side of this axon. So, for example, if there's another neuron on the other side, and this neuron is here, then this middle neuron may be activated by either one of those or both of those. So this is something also very important to understand, that these neurochemicals in general also add another level or another layer of statistics into this entire topology, entire flow of information. This is very important to explain. So here we have a much more complicated. Now this is still a very tiny example, but this is showing a cluster. So starting from the left, we have a neuron here which flows into another one here. This flows into this one here, and then this flows into three neurons, which goes into this light chain here and another longer chain over here. Okay, and then this neuron has another chain, a long one here. So this might be, let's say, the corpus callosum that goes to the other half of the brain. Okay, let's say this is 6 or 7 inches long, and then this is on the other half of the brain, and then this terminates here, and then this activates a set of neurons here and it activates a long set of neurons here. So this is a more realistic example of how neurons are. Although in the real brain, animal brains and human brains, we wouldn't have just here, we have roughly about 15 or 20 neurons. In a real brain, there would be, if we were just talking about a single cluster would probably have 50 million neurons. We're talking about huge numbers, cluster of clusters. And a small cluster might have, let's say, 1 million neurons. So it's very important. And the level of clustering and the type of clustering is also very complicated. I'm going to describe one more thing. So again, information still flows in one direction. Okay, it flows here, although it's not as simple anymore, because now from this neuron, it flows from left to right. But if you see this connects to 3 different neurons. One of these neurons is going north. The other neuron is going west again, right? It's not going to the right. It's not going eastward. It's going west. It's going to the left. So it's, in a sense, the signals are always still flowing from the cell body of the neuron to the terminals. It's always going in that direction. So every one of these neurons, it still flows in the same direction relative to the neuron. But in terms of the topology, information can flow anywhere based on how these neurons have grown, how they actually sit within the brain. This is what I'm saying, is that it's no longer flowing necessarily in in a linear direction. In general, roughly you could say it's going left to right, but it's also going bottom to top, and it's also in some cases going from right to left. And what I did here is I just added, there's a few more neurons that I just added here. There's a cluster here, there's a cluster here, there's one here. Down here, there's a neuron, and up in the top left corner, there's a neuron. I just added, so if you can't see, I'm just kind of toggling these so you can see I added a bunch of new neurons. And the reason why is because typically in the brain, not every neuron is connected to other neurons. Sometimes there are neurons that aren't connected, or sometimes they're connected and they have their own network. So this neuron here isn't connected to any of these, but it might be connected to other neurons and form a different cluster. Okay, so this cluster of cells has no connection with this neuron here, and the cluster that it's tied into, it might be going off in a long branch, which I haven't shown here, but it might be going off in a long branch to 20 different other neurons, or a million different other neurons. But these sets of neurons down here and this neuron and this group have no interconnection between them. That's what I'm trying to explain here, is that, and there are many, many types of neurons like this, and there are many, many types of clusters, which physically they are adjacent and nearby each other, and they actually mesh within each other. They actually, sit within each other, but they may not have any connection just because of how the neurons grew. Now, one of the videos that I linked to in the talk, if you didn't get a chance, I'd recommend the fourth video, actually goes a little bit into this. That video talks about the connectome of the brain, and what the researcher, the scientist, describes is that what's important is that in a typical brain, human brain or animal, any type of brain, it doesn't matter how these neurons grow. These neurons basically grow randomly. They grow in random directions. When an infant is born, when from a zygote to a fetus, etc., and the fetus grows and the brain cells, stem cells become brain cells, and neuronal cells split, etc. All of these cells that the 100 billion cells in a born infant that develop, they all basically grow randomly anywhere and everywhere. There's no rhyme or reason for it. That's what's important. Now we've learned that there is realistically no specific location for every neuron or any neuron, although there are patterns in that statistically and even topologically. So the cortex of our brain, the topmost layer of our brain, is a few millimeters thick. It's about 5 millimeters thick, and it actually consists of 6 specific layers. And each of these layers have different types of neurons. So that's an area where neurons do grow in specific ways. And also the fact that we have a corpus callosum, every human pretty much has a corpus callosum, means that we do have these very long neurons which do grow. And those are programmed through our DNA, through our genetics. The fact that we're human beings, and the fact that we are mammals and reptiles and all of these organisms, early on in our evolutionary tree, the development of corpus callosum came about, and our genetic DNA added all of this into it. So that's where all of that is. So these are the cells that I added in, which don't have any connection to the main cluster that we're talking about here. So we're almost at the end of this. I'm going to have a question answer after this. So these are 5 different snapshots. Again, these are cartoons. These are not actual. After that, I have actual image of what a representation of real brains. On the left is a type of structure that exists in a newborn's brain, and this is a tiny, tiny dot of an area. And what you'll see is that each of these, there's a large black dot with a long axon, and most of these are running vertically, right? And that's because initially when I talked about those layers in the brain, most of those layers are running vertically. And so a newborn's brain has all of these layers vertically oriented, although some do run horizontally for other reasons. But what's important here is that the newborn's brain actually has more neurons than an adult's brain. That I want to say, that's the reason why in the title I say density, not quantity, because the density increases from left to right, but the quantity actually decreases from left to right. This is important, and this is kind of counterintuitive.
Could you move the overlay at least so we can see how they're just up a little?
This is just rough. So a newborn's brain, 1 month, 9 months, 2 years, and an adult brain.
Thank you.
So these are just rough time frames, and at the top, the what it says is a density, not quantity. So what's important here is that from going from left to right, this is showing the density increases, right? You can see this on the right here. It's so dense. You have all of these little lines in between. And what this means is that the interconnections between these neurons are much more. Okay? Although the actual number of cells, the number of these dark splotches have reduced. So the number of cells reduces as we grow older. Okay? But the interconnections between them increases. And the interconnections are actually where learning happens. This is the reason why it happens, because the learning, and this is a physical version of what I said earlier at the beginning, that information flows and also it's stored between the cells, between the neurons, because these axons are actually what encode the stored learning that an adult brain has, or a teen brain for that matter has. It encodes the learning that we have. So that's very important to understand. The next slide is, this is an actual real neuron, although this is not a photograph. This is it's a rebuilt construction based on a physical, actual human brain. Actually, no, I think this was not a human brain. This may have been a mammalian brain, but it's no different in human brain. So this is the cell body, which you'll see, and these fibers are basically the dendrites. In this case, let me explain that because of the type of imaging that they were doing here, this dendrite actually will have many, many finer hair-like projections. It won't have just these roughly 10 that we see here. It probably will have several hundred. But because of the type of imaging that was done here, the imaging system was not able to capture the extremely fine level of dendritic tree that exists here. So it only captures the really thick branches. But if you think about each of these thick branches, and there'll be dozens and dozens of more fibers coming off of this and coming off of these thicker branches that will form almost like a tree canopy here on this side. And then all of those will be waiting there to pick up signal from neighboring neurons. So there will be another, surrounding neighboring neurons all around it, which will not connect physically, but connect, proximally nearby. And the signal can flow from one or more of those neurons into this one red neuron. And then you see this thick, projection outward. And there's a purple section in the middle here, and that's more because there's a video that this is around, and that video wanted to show the fact that neurons exist with other types of cells. This purple section, I don't show it here, but this purple section contains many, many other types of non-neuronal cells also. Some of those other cells are smaller. Many of them are much smaller, and they're support cells that help a neuron to survive. For example, they provide oxygenation, nutrients to the neuron, just like any other cell. They provide temperature regulation, all kinds of other things. So that's what that purple thing in the middle is. But basically, this long path is the axon, and it goes off far away. And what you'll see is that this axon itself has certain branches, and this is true. They actually have branches. So depending on the type of neuron, this is a simple neuron. It has one long axon. Some neurons have basically two axons. Some neurons are like a T-shape. They have an axon that goes in two opposite directions, and they can sometimes have a third axon. That's why they're called theta. So anyway, this is a picture of a single neuron. This is a representation image. This is a real representation of many, roughly 10, 15 neurons. And this is how tightly they exist within each other. Now, what's important is all of these might not physically connect to each other, depending on how these dendrites and axons and axon terminuses connect. They might or might not. So in this case, the soma, the cell bodies are so close to each other. And although here, this yellow one here, you see, the axon is very short, right? So it actually ends just where the blue one is. So this might be one where this yellow one actually does connect to the blue one because it's so close to it. But this orange one here in the middle might connect this yellow one. It might actually go further and connect to additional neurons up here. But the red one that's in the middle here, the one that we saw earlier, its axon basically goes down and it doesn't connect to anything else here. So this doesn't connect. So when you see a clump of neurons, the connection between them can be very complicated. And today, we don't have an ability to understand the interconnections by looking at actual brain slices or anything like this. It's impossible for us to tell today. Although topologically and in terms of a simplified model of it, we can do that. And that's typically what we do. So we're almost at the end. We're gonna go into Q&A now. So let me stop the share, and we have some questions in the chat.
Okay. Well, why don't we start with the live questions? Go ahead and type exclamation point or raise your hand in
So I think people
Zoom.
already asked questions. Do we want to go with their questions first?
Sure. I mean, are there any questions in particular? There are quite a few. Some more than.
So I think somebody asked about speaking about the corpus callosum. Nicole did. So I answered. And Nicole, is there anything else you want to know? We're not going to go into a lot of detail with brain physiology, but the corpus callosum is a set of nerves which connect the left and right hemispheres, and it's our band.
I just want to know why things seem so complicated when they shouldn't be, and things seem so frustrating when they shouldn't be.
So what do you mean by complicated and frustrating? Do you mean in terms of how we behave, our behavior, or how the physiology of the brain looks? Do you mean
how we react?
Okay. Why that's so complicated? Well, that gets into much higher level functions. So right now and again, I mentioned earlier that we're going to talk about small clusters of neurons here. Small clusters of neurons don't really create our behavior. Okay? Our behavior is created by large clusters and large groups and information flow between brain regions, which we're not going to go into tonight because it's a much more higher level concept. I've gone into some of that in prior talks. So if you want to answer, I'd recommend you go back and look and find some of those that might, touch on those.
Thank you. I'm not trying to be frustrating to you.
Thank you, Nicole.
No, it's a fair question. I want people to ask questions like this because this is, we're trying to learn and we're trying to understand. It's a valid question, but I can't go into too much of that right now. But specific to the topic that we had tonight, that's what I want people to focus on. Because people have questions on anything and everything with the brain. We can't go into all of that. I might touch on here and there a few things that relate, but if your questions are about complex behavior, we're not going to go into that tonight. I've actually done talks in the past around that you're welcome to look into, or in future when we come back to those, we'll go into those again. So next, I think.
So why don't we go to Vanessa next, actually?
Okay.
And folks, go ahead and type exclamation point in the chat. I actually have a question too, if you don't mind.
Okay. Yeah. I've got two quick questions. You say can only go one direction. What happens, let's say if there's a log jam where it's sending out so much information, but it's not being absorbed by, I guess, the next neuron. And the other thing is, let's say there's 5 neurons firing to the same one, 5 different pieces of information. And then let's say another one is getting 100 pieces, but let's say 4 of them are the same information. Do the 4 of them get priority, or are they more likely to be emphasized or treated differently? Does that kind of make sense of what I was saying, or?
Yeah. So the second part, or the latter part of your question about the prioritization, that's very complicated. And the word statistics, I'm not going to use it, but basically think of it as a pattern, right? So the pattern of how close they are, right? The axon terminals of all of the other neurons that are coming in, the input neurons, how close they are, the level of closeness, whether they use the same type of neurochemicals or neurotransmitters or not, because some neurons use certain types, other neurons use different types. So if the input neurons are emitting one type of neurotransmitter, but the receiving neuron doesn't use that, information won't flow, right? So that's another level. So the whether information flows or not is actually very complicated. It depends on many, many factors: the density of neurons, the direction orientation, the types of neurons, whether they're excitatory neurons or inhibitory neurons. That's something I roughly mentioned, meaning that some neurons, when the information flows and they reach the end of the terminal of their neuron, when neurotransmitters are released, those neurotransmitters actually turn off the next neuron. They don't turn on, they actually turn off the next neuron. So if you understand what that means, if you have a neuron and it has all of these inputs from other neurons coming in, right? Let's say it has 5 neurons that are inputting. Out of those 5 neurons, if 2 of those are inhibitory, meaning they inhibit the signal, and 2 of other 3 are excitatory. So when all 3 of those excitatory signals are on, right, and the inhibitory ones are off, then the signal will flow, right? But let's say all 3 of those excitatory, the ones that are exciting the neuron, if they're on, but the 2 inhibitory ones are inhibiting it, they're stopping it, right? So even though the 3 that are exciting it, they're on, the other 2 that are inhibiting it, turn it off. So this adds to the level of sophistication and complexity in a single neuron, that the information that's flowing, it's almost like you have a switch, right? That even though those 3 that are sending a signal in, those other 2 that are inhibiting it, they're acting as an off switch. They're turning off that neuron in a sense. So it gets very complicated very quickly. So Vanessa, your question about log jams really don't occur. Or in another way to look at it is that when a neuron signal doesn't flow through, you can think of that as a log jam. It's not that it's a log jam, it's just that the signal basically dissipated, died off. The signal doesn't move forward, it doesn't propagate. And that's, in a sense, that's by intent. If you want to think of it as all of the complex things that happen in a brain, right? If you want to think of it at the most minute, simplest level, let's say that you have a reaction. Let's say that the neuron we're talking about is in our visual field, right? And our visual field sees something flowing across. Let's say realistically it's a ball, okay, flowing across. But our brain doesn't know it's a ball because that physically takes time for our brain to register and react and understand that it's a ball. Initially, it first sees something is moving, and our brain registers movement initially, very, very quickly, just like that, it registers movement. And initially, that reaction might flow throughout our brain. And if that movement is on the ground, our brain might think that's a snake moving very rapidly, right? So that signal might flow all throughout our brain. It might go to our amygdala. Our amygdala might be activated initially by just simple movement, okay? Because it doesn't know if it's a snake. It doesn't know if it's what it is. Okay, it always assumes the worst in a sense. But because of these inhibitory neurons, pretty quickly, the amygdala gets turned off. Because the region of our brain that does processing of what that thing is that's moving, it recognizes it's a tennis ball. And once it recognizes a tennis ball, it sends inhibitory signals to other parts, which turn off. So for example, it'll send signals to the amygdala to turn off our amygdala so it doesn't get excited over this tennis ball.
Yeah, thank you for that answer. So it looks like we have Ella and then wants to ask a question and maybe be a little bit shorter on the answer because we have to get the second part of the second part of the presentation. I do want to ask about neuroplasticity at some point, but I think that, that's directly related to this. Go ahead, Ella, followed by Ron and
yes, can a neuromuscular malfunction, this specifically I'm asking on the nerve side, not the muscular side of the junction, the nerve side. Can a neuromuscular issue on the nerve side with calcium channel high antibodies are indicated cause issues other than muscular weakness, numbness? Can it cause issues such as cognitive issues, heart rate, and balance issues as well? Because we're discussing how these neurons are grouped together and how they might affect one another. This is just going into a more specific question. And are these neuromuscular neurons with others as well? I have it in the chat. I just didn't put that it was on the nerve side that I was more interested in.
Thank you for that question, Ella.
Yeah, so just, so I'm not a medical doctor, and we don't typically go into medical questions. This is not a medical question, so I will answer it. But my answer will be, so before I answer that, let me explain that. Typically, when we're talking about signal transduction within the body, within the brain and body, rarely is there just one single neuron or one single axon that carries a signal. So what I mentioned earlier is that nerves are actually multiple axons that carry a signal. So what that means is that there are actually multiple nerves. Typically, there are hundreds and usually thousands or millions of nerves that together can form a bundle. And the nerve bundle can actually have nerves which are very different from each other, or they have different function. So, for example, when you have a nerve that's carrying the signal toward a muscle to activate that muscle, let's say, right? Some of those nerves, some of those axons may be inhibitory axons. Some of those may be excitatory axons. That adds another level of complexity here. So in general, and we can't say exactly what, I'm not giving a specific example, but in general, depending on the overall pattern of signals that flow through the nerve to the muscle, depending on the types of nerves, depending on the types of neurons that they are, And again, depending on the types of neurotransmitters that are used. So what I said earlier, as far as signal flow from neuron to neuron, same thing applies for signal flow from neuron to musculature, or from sensory neurons or sensory receptors to neurons, the input side or the output side or the between side, neuron to neuron. The same thing applies to any of these areas. So hopefully that explains what you asked. But basically, the dysfunction can be from many different reasons. It can be that actual physical neuron nerve bundle has been damaged. If you're talking about specifically MS is much more complicated. The genetics causes a degradation of the nerves themselves. The nerves don't grow properly, and also the way signals are flow within the nerves. Again, I'm not a medical doctor, so I can't give you details of that. But my understanding is that the signal doesn't flow properly, as well as there is actual structural abnormalities in these nerves that cause problems. And the nerves die over time.
So really quickly, there seems to be a brief question by Dwayne in the chat. Roughly speaking, what is the median and mean number of axons per neuron? Do you know that offhand?
I don't know. I don't think anybody can give you a real answer on that.
Okay.
I mean, the rough. So when you're talking about median versus mean, median in terms of, because the median would actually have to be a more explained statistic. I'm not going to go into statistics here, but you asked statistical questions. So when you talk about median, for example, if you talk about the distance between the terminus of one neuron and the dendrite of the end of the dendrite of the next neuron, that distance can be used to form statistics. So you can come up with a median distance between the number of median distance as well as the number of connections between neurons. So median, there's no such thing as one median. You can have multiple types of medians when you ask this question. The same thing for mean, although mean is a little bit easier. But nobody knows. There's no one answer. In general, when people talk about it for human brain or most large mammal brains, it's in the order of tens of thousands to millions. And again, depends on specifically what type of brain region we're talking about. Some brain regions, the neurons don't have a lot of neighboring neurons. Other regions, there are tons and tons of them.
Ron.
Yes, I have a question. I'm very confused. I've been reading recently at a layman's level, left hemisphere versus right hemisphere. They both process data. I assume data that's simultaneously happening and it's being processed and very differently. So in everything that you've spoken of today, is there some simple way that, for a layman to say, why is it that the left hemisphere will process it and come up with information differently than the right hemisphere? Is it because the neurons in the two hemispheres are they the characters that sort of make the difference between the way the left hemisphere sees the world and the way the right hemisphere sees the world? Or
Okay.
You see where my,
Yeah, I understand. So the importance of why there may be different types of information processing in the two hemispheres, right? And whether they encode different information or how they integrate together, you're asking that type of question.
Yeah.
So before I answer, let me give a little bit of understanding of the fact that we have two hemispheres and why that's important. So I've talked about this in many prior talks. We as human beings, we're animals. We've evolved from other animals, and our brain has evolved from other animals. And the fact that we have two hemispheres evolved for a very specific reason. It's because if you look at some animals in the world, right, for example, birds or horses, the many specific types of animals, they basically, their brain, half of their brain sleeps at one time and the other half of their brain sleeps at another time, okay? And this is what allows them to survive, because horses are not predators, okay? Many birds are not predators. Birds can be both predators and prey, but birds need to fly, and birds need to stay in the air even when they're asleep. So the reason why birds are able to sleep when they're flying is that half of their brain is sleeping while the other half is doing the flying. and then it flips after a certain time. This part that's sleeping wakes up and it picks up the flying activity, and the other half goes to sleep. Okay? So this is important behavior for a bird. Similarly, for a prey animal, it needs to when it's sleeping, half of its brain is sleeping, and the half of the brain that's sleeping, and again, remember that the body crosses with the hemisphere. So the right side of the body, all of the signals and information from the right side of the body goes into the left hemisphere and vice versa. So in a horse, the right eye will be open and will be alert to what's happening on one side, and the left half of the brain will be awake. But the right half of the brain will be asleep, and the left eye will be closed and sleeping. And that's how a lot of animals actually sleep. And this is biologically one of the reasons why we have two hemispheres, because it's necessary for survival. Now, we humans don't need that anymore because we're predatory animals. We don't need that, okay? But evolutionarily, that aspect stuck with us and it's still in us, okay? So this is why we have two hemispheres. So there's no real purpose for us to have two hemispheres. Our two hemispheres don't really inherently, they don't do a whole lot of cross-pollination. It's almost like we have two different hemispheres that work independently. They do share a lot of information. And now in our modern brain, when we understand the world, there is information that flows between them. But if you look at certain types of diseases where the corpus callosum has been severed in certain people, outwardly they function as well as most other people. You can't tell somebody's corpus callosum has been severed, but when it's severed, very specific things happen in those people. Is that they lose a type of information flow that normally happens in a person, but very little information, very specific, but little information flows between two hemispheres. So your question really, it's almost as if it's not really important how much information flows between the corpus callosum, because most of the information that's in each half stays within each half. Most of it doesn't flow to the other half. Okay, each hemisphere is pretty much acting as a dependent part of itself. Okay, and what helps us, what proves this, is the way our visual system works. The visual system in most mammals, actually in most reptiles and even lower form animals, is that the way our brain eyes work. I'm not going to go into this. There's a talk I did earlier. I think two talks.
Yeah, I was going to make a suggestion actually for this, instead of going into detail, is that Ron and I will look for that meetup in the chat where Sanjay actually covered this topic in great detail and gave us a set of videos that we could look at. So I will look for that as we go on.
Yeah. So just
can I ask one really short follow-up question? Very short.
Let's not have follow-up, Ron. At the end, if you want to, after other people, I'm sorry, but I want to get through because a lot of people, I don't want to have people waiting. Okay. But I do want to say, Ron, the reason why I'm bringing up the visual cortex is that that's a very different area than rest of our brains. The visual cortex actually mixes the information completely from the beginning. It's very integrated. This is the reason why we have stereo vision. Right, so other than that, a lot of information doesn't really, it does flow. There's information that flows, but there's not a whole lot,
So I have a really quick question around like how our neurons impact our structural plasticity. Like, so the idea that, how does that, can you go into detail just a little bit, how that actually functions and how that impacts not only our structural plasticity, but our functional plasticity, and how our neurons interactions, actually the chemicals in our brains impact that, just briefly.
So so plasticity basically is the ability of our brain, or any brain, to change its wiring. Okay, and change its wiring means several things. It means that, and this happens through normal biological processes. It also happens through the way we learn. So neurons die. They don't always die. They rarely die, but over a lifespan, they do die. And especially in youth, a large number of neurons are intentionally killed off. They're intentionally pruned. Up to about age 7, 8. We start to lose a tremendous amount of children lose a tremendous amount of neurons. They go from roughly 100 billion neurons to around 86 billion neurons, again, on average. So 14 billion neurons, almost, 1/7 of the brain, 1/6 of the brain is destroyed. Literally, the brain destroys itself. And that's the reason because it allows us to learn more. Because if the brain is overcrowded, there's too many neurons, the level of learning and the amount of learning and how easy it is to learn actually gets inhibited. So this is something that happened through evolution, that our brain does this automatically. Our genes make this happen within our brain. So plasticity is this pruning, meaning getting rid of specific neurons, the entire cells, right? It's also with the dendrites and axons. You can have pruning of the dendrites within a specific cell. That cell, because of things that are happening around it, because of the biochemical nature of things around it, it's sitting in a bath, you can say, almost like a soup of neurochemicals around it. And those neurochemical soup affects each and every neuron and how it functions, how it grows and develops, and whether it elongates or whether it grows in one direction or another direction. All of these things are affected and controlled by the chemical soup around the neuron, as well as the support cells, other cells that are around it. So neuroplasticity is very complicated. So the level of cell death, the level of pruning of dendrites and axons, the level of direction growth, all of these things are aspects of pruning. And when we're talking about growth, when we're talking about an axon growing, okay, that's actually where learning happens. When we learn something, when we form habits, anything, as children, when we learn the alphabet, as adults, we learn to drive the car. Any type of learning, even when you remember the dream that you had last night, that's a type of learning. Any type of learning actually forms connections. And, even though the connection might be, very tiny, there is a level of growth and there is a level of activation between. There's something called LTP, which I'm not going to go to today, but LTP is a specific mechanism which allows the two neurons to quote unquote connect. They don't physically connect, but they connect in an energy flow way. It's a little hard to explain, but they connect in a way that the probability of information going from one to the next increases. So in general, plasticity, and this is plasticity. And then depending on how a specific neuron and how the thousands and millions of neurons around it grow and develop and change over time, that affects how the functions that those neurons together perform in our body, in our brain, how those functions get changed. So if all of those neurons are specific to a specific memory, right? The memory of the car, somebody gave an example earlier. And if the dendrites are getting pruned off, well, that memory of the car will fade away. Or if the axons grow and connect into more neurons, then the memory of the car either might become stronger or it might evolve into a memory of a different car. Like, the memory might be of a childhood car that you had when you were growing up, and it might connect into a brand new car that you bought as an adult. And so your childhood memory of a car may actually merge into the memory of a car that you just bought. So, when plasticity happens, this is why, because when growth happens, growth can happen into areas that connect into different ideas or different memories or different processes. They don't have to be the same process. That's why memories can merge.
Yeah, that's actually interesting, because I also want to come back, well, if we have a chance, come back to some of those things, specifically, the environmental aspect of it that you mentioned, the developmental aspect of it, I think is interesting. And you've talked about some of these things in the past as well. So why don't we go to the second part of the presentation right now? And we'll come back to any questions there are in the chat. There's one more left that I think is really important to get to, but we'll get to it, I promise you, Dwayne, at the end. And then let's get to this next set of questions or next set of slides. And then how long do you think will you need for this next?
I can wrap this up fairly quickly.
Okay, all right. So
I don't have a lot of slides. Mostly it's going to be talking, but I can wrap it up if we need.
Okay, well, let's do it. Let's do it.
So maybe 10 minutes, maybe at most, I think.
Okay.
Right. So let me start. So hopefully everybody can see the my Zoom is I don't know where my Zoom is. Sorry, I'm trying to. So hopefully you don't see the. Is there anything blocking?
No, it's actually good. So thank you very much.
All right.
Whatever you did.
So I changed the video. So basically, last time I had it for video, so I removed those options. So I should be better. Okay. So now we're going to go into a little more of the statistics. So we started talking about this a little bit, but first off, let me talk about there's a neuroscientist last century, mid-century, Donald Hebb. He actually had an idea which is now today called the Hebb's maxim, It's basically that cells that fire together, wire together. And this is a very simple idea that some of you may have heard of, but it's very important. And this is basically at the heart of what we're talking about tonight, is that cells, so for example, in the diagrams that we had, when a single cell sends a signal through, and in a coming up slide, I'm going to have an example of what happens when cells actually fire, when a neuron actually fires, what the signal looks like, although we're not going to go too much into it. I've talked about almost everything that we've done tonight. I've talked about these in prior talks. The difficulty is that, I can't really give you a specific single talk because, there's a large area that we're talked about. But there are specific talks about neurons that we've talked about. There's specific, for example, we talked about vision. So there are specific topics that we've covered. So if there's something that you want to understand better, you can go to that, or in a future talk, you can raise it and we'll try to go a little more into it. Although in each talk, we try to stick to the topic that's at hand. But if there are some peripheral questions that, we do entertain, and we go into those if they help the discussion. So cells that fire together, wire together. Wire together means they grow together. This LTP activation that I talked about, LTP is long-term potentiation. It stands for long-term potentiation, which basically means that when you fire a signal from one neuron into a second neuron, into the target neuron, over and over and over and over. So you do that long-term, right, for a long time. What happens is that signal that keeps passing through the first neuron into the second neuron, it almost like it carves out a path. Now, it's not physically, but if you think about it in terms of the topology, it's almost like it creates an easy path for the information to flow from the first neuron into the second neuron. And that path remains. Those two neurons stay interconnected for a long, long time. Now, that interconnection can be weakened if there are other neurons, because remember, neurons are crowded around. It's a very crowded place, and there are neurotransmitters that are flowing all over the place. So those neurotransmitters may cause other neurons to grow into that area. And the number of neurotransmitters that pass from one neuron to the other is a very important part of how information flows. So the fact that these two neurons have learned to fire together, right? They've been firing together and they become wired together because the neurotransmitter that goes from the first neuron, most of it goes into the second neuron. But now let's say in the future that in that synapse, in that point between them, other neurons grew into there. This person grew, they became an adult, and other neurons grew into that area. So those other neurons now are also going to take up those neurotransmitters. So all the neurotransmitters that the original one neuron is sending out, all of those don't go make it into that second neuron. Some of it get diverted out into these other neurons that are crowding it out. So in that case, the signal connection over time has weakened. But if the person keeps doing the same thing, if they keep firing over and over, for example, let's say these two neurons have to do with speech in the English language. Well, if that person keeps speaking the English language, then that connection will again get stronger because they keep speaking the English language. But let's say that this is from a language they learned in their childhood and they no longer speak it. Well, the fact that these other neurons came in and that original signal is now weaker, this is an example whether you're starting to forget a language that you knew in your childhood. And so these other neurons that came in probably have come in because you're speaking English and you forgot French because you learned French in school and you no longer speak French natively now, or you don't speak it frequently enough, so you're starting to forget it. So these other neurons that grew into that spot are the neurons that have to do with your English speech. So they're going in because you're strengthening your English skills, and the skill with your French language has gotten weaker. So this is an example. And this is also an example with the plasticity that Joe asked about. So cells that fire together, wire together, this is more important. And there's a corollary to this, that the more often they fire, the better they wire. And this is very important also. So this is a video that I have, which is going to show, so this is actually, so this is showing what actually happens in a neuron. Now, what's happened here is there's two graphs that you're seeing, and let me just explain quickly. So this is a single neuron, okay? And this neuron, this here on the outside is the axon. If you see, it's turning purple, okay? And the neural cell body itself is turning purple. Now, what the researchers have done here is that they've actually developed a very special molecule which fluoresces, it glows, it literally glows, it glows purple. And they've built this into this neuron so that whenever this neuron activates, it literally glows. So this video is of a real live neuron. And this is, I'm sure it's an animal because we can't do this in humans. It's dangerous. So in an animal, it's probably a very, very basic animal like a zebrafish or something, a very primitive animal. Nonetheless, it happens the same way in even human neural cells. So this neuron, when it gets activated, the entire neuron flow, flashes, because this special molecule called a Voltron molecule, it's a patented molecule, but it's because it's used in research. It's a very powerful molecule. It helps us see that this neuron is firing. It also helps us see that the axon is also carrying the energy. So what this molecule does is it shows the voltage. So wherever it's turning purple, there's actually ionic voltage happening there. So the ionic voltage is happening all throughout this axon. So these two graphs, the purple is showing the light intensity. It's inverted, and the white on the bottom is showing the actual voltage. So what you see, what this is showing is that when a neuron fires, this is the reason why I said earlier, it's not like electrical current like in electrical circuits or computing circuits. In a computing circuit, you'll have a current that flows continuously. Here you have pulses. When an electron fires, it actually pulses very rapidly, and these are microseconds apart. They're actually firing, it's a very rapid succession. If you hear this in real time, it's almost like it's a rapid fire buzzing. It'll sound like a buzzing because it's happening so quickly to each other. So this is a real picture and this has been slowed down. So this is actually happening in about a second and a half. This entire activity is happening within about 1.5 seconds. So it's flashing about, about 18 times within roughly 1.5 seconds. So it's in this case, it's several milliseconds apart. So this is a slowed down neuron. This may be a neuron that's in a peripheral system. But basically, this is an image of what actually happens in our brains. When signals fire. And so this is not showing the successive neuron, but this axon has two forks to it. There probably are other neurons that will be activated depending on many things, statistically many things, but the information is flowing from the cell through the axon into these axon terminuses and probably into other neurons. And the information is coming, if you see these dendrites on this side, it's coming in through these, one or more of these. We can't see how it's coming in because there's no voltage there. Those actually come in through neurotransmitters. They're not carried in through voltages. That's why you don't see a lot of flashing going on. The flashing that you see up here is actually a reflection off of the neuron. It's so bright up here. Anyway, so this is a real image of, we have the same image that we had earlier. What I want to talk about here is that we have our original, so this is just to remind you that we have this complicated, in this case, it's a very simple model again, but this complicated network that we have of images. So if you think about the pulses flowing, when the pulse flows from this neuron to the next, it's not that the signal flows in a way. The signal pulses rapidly and only does it for a short period of time, maybe a second, or it could even be several milliseconds, depending on the type of signal, depending on the intensity of the signal. In this case, signal is coming from only one neuron. So chances are that when this neuron activates, it won't fire for too long, or it won't be for very strong impulses. But if you see over here, this neuron is getting its. Actually, let me find one that has multiple inputs. Actually, I don't think I have any here that have multiple inputs going into it. Most of these have single inputs. But these two here might actually have inputs from itself, because if there's enough neurotransmitters emitted, they actually can activate itself, which would be, it could be kind of a problem, but it could be part of a feature of the activation. But anyway, the way that these neurotransmitters pass from cell to cell, and the way that the electrical pulses, ionic pulses, flow within the axons, these are all aspects of this flow, information flow within neurons. So we're starting to wrap up. So basically, information flows in the form of ionic signals and in the form of neurochemicals, specifically neurotransmitters. Those are the most common, but there are other types of other chemicals. Ionic signals, they take an exact path within the axons. The axons are those exact, almost like wires, right? So they take those paths, and the signal is fairly precise. If you saw the graph earlier, the pulsing, it's a very regular, precise signal that passes from one neuron and goes into the next neuron. And if the next neuron has multiple inputs like that, all of them, then chances are that it will activate the neuron much more likely. So because of this aspect of precision, the ionic pathway is much better. And that's the reason why our nerves are very important in biology, because our nerves, this is basically the nerves. Our nerves are very important in our body's function. And they are important because they are a very exact way of passing signal. They're precise, they're very, very fast also. I didn't add that, but these are extremely fast. They're not as fast as electrical signals in electrical circuits. They don't travel at the speed of light or close to the speed of light as electrical signals do, but still they're relatively fast compared to this other method. Now, the other method is neurotransmitters. They're not exact. They float around. They form almost kind of a smoky, or I use the example of a perfume. They pervade the area around it after they're released from the axon terminus. So they're not very exact. They kind of fill the space around the end of the neuron. They can go to the wrong neuron or even to non-neurons. They can go to other cells that are in the area. And some of those other cells do absorb these neurotransmitters. And they use them and they recycle them. And sometimes they convert them into other neurotransmitters. And then they get absorbed back into the same neuron, or they can be absorbed back by other neurons, neighboring neurons. So neurotransmitters are much more complicated. So the transfer of information is not very precise, is not very exact. Signal is sometimes imprecise, and in that sense, it's statistical and random. The level of statistics and randomness in the information flow is mostly because of these neurotransmitters and because of neurochemicals in general, and because of the complex nature of how all of these neuronal cells inhabit the same space. There's so many, thousands and millions of them. The fact that these axons and dendrites and terminals are sometimes far apart, sometimes close together. Sometimes they grow toward one, sometimes they grow toward another, sometimes they grow in one direction, and then they change course after a few months, they go in a different direction. So all of these factors together come back to elicit the statistical nature of information flow. Now, another thing that, so in this example, if this neuron was activated, the information would flow through here, and then they would go through here, and they would, so in this case, this, after it reaches the end of this terminus, it actually splits into one path here, another path leftward, and one path going up, and another path going leftward. And then this path leftward goes into these two paths here. It follows forward, So this signal actually goes into two different areas, and then the second direction also continues, and it will go off into other directions further. So this one neuron can actually form a cascade of two different or even more. Typically, if there's thousands or millions of connections between, one neuron, when it gets activated, can actually affect thousands of other neurons around it. And it can affect neurons far away. This chain I ended because I didn't want this to be too crowded. This chain typically would continue for hundreds and hundreds of other neurons forward. And so what you'll see is this signal, and then each of these signals might split even further. So this signal here might split into 3 neurons, would split. So the signal and the probability of this one signal actually taking one path and not another path, that's another area of statistics that we're dealing with here, is that this signal starts off very solidly here. It's a signal, and whether this, remember, this signal can be an excitatory signal, meaning it's turning on the neurons on the downward stream, or it's turning them off. It can be an inhibitory signal. So either type of signal, whether it's inhibitory or excitatory, it can go into both of these directions initially, or maybe one, depending on how if let's say the distance between these two was further apart, the chance of this activating the second pathway would be a little bit less because the distance is further, and it would more often activate this. But in this example, the way I've drawn it, it activates both of these more likely, so it's almost 50-50. But then after it goes a few neurons deep, this signal, the chance of it actually activating further signals gets less and less, and it actually starts to follow a statistical process. And the same for this other second pathway, it starts to become very statistical. So the fact that this signal starts off with this one neuron, and whether it actually activates a totally different cluster somewhere far away, if you want to look at the probability of it activating another cluster, it's very difficult to calculate. And that's why we're not at that level in neuroscience to do that. That's probably decades away. So this is another level of complexity, but this is the real thing that happens in neurons. Now, and then this grows. So there's another thing that I added here with all these red dots you may see. And these red dots are basically symbolic of the neurotransmitters. They basically are emitted out of the axonal ends of each neurotransmitter. And what you'll see is that some of these neurotransmitters kind of, if there's a lot of them, they kind of get emitted and they start to float outward. And so this neuron here is emitting all of these neurotransmitters, and some of them get close to this next neuron and they activate it. But some of the neurotransmitters actually float around, and they actually may start to reach even as far as this neuron, even though it's very, very far. If the signal for this neuron is very, very strong, or if it's repetitive and very if it pulses very, very frequently, then it may emit enough neurotransmitters that they start to reach this neuron, and they might actually accidentally fire off this whole branch, which might be a branch that goes off into another, million neurons, which is normally is not connected at all. But because of an accident, because of these neurotransmitters, the proliferation of these neurotransmitters at a specific time, moment in time, let's say that you're dreaming. So this is an example of hallucination, where what might be happening is that when you're awake, the level of neurotransmitter activity will be sustained to a certain level. But dreaming, you may actually emit more neurotransmitters that actually go into other areas, that go into other connections that actually aren't connections. So this might be a hallucinatory pathway that makes you think of something weird. Like this neuron might have to do with the car that you're dreaming of. And this pathway might have to do with flying. And so when you're dreaming, this might connect the pathway of a car. And then when it's activated with this, you get the sense of a car that's flying, right? And all because you have excess neurotransmitters when you're dreaming. So this is an example of the complexity of what happens in our mind because of the distance, because of the number of neurotransmitters. Again, I only drew one type of neurotransmitter. All of these are red dots. There are 30 types, and typically in any given neuron, we have between 2 and 5, 2 and 4, 2 and 5 types of neurotransmitters. So if you add in multiple types of other neurotransmitters into this mix, the complexity of which neuron activates which other neuron gets really, really difficult to understand. But if you simplify this into a model, you understand what's in general happening with our brain. And we can't use that to predict what will happen in a specific person because we don't have this map. We don't have the ability to measure a specific, at least in a human being, a live human being, because it would be damaging. So we don't do it. But in animals, we do this. In animals, we can study specific neurons and their activation, how they connect to other neurons, so we can do it. In animals, for example, if one of these neurons connects into a motor circuit way, which let's say this motor circuit way made them move their thumb, we can actually force this neuron to activate, which would cause, or let's say not that, let's say this long neuron goes into this pathway here, and it actually goes into the thumb. So by activating here, we can actually see what types of situations would all of these things happen where the thumb actually moves based on this neuron activating. Or you might activate this neuron and suppress some of the other neurons in the middle, turn them off in the middle and see what happens. So we can actually test out this complicated circuit in an animal to see what's happening. And these are the types of things we can do to some extent. In very simple animals, we can do this because this, to some extent, it technically doesn't harm, but it is invasive, and we don't do this in people or even some types of animals. So that's actually the one last slide. So information flow. So the flow of information is statistical. It depends on the proximity of neurons. So how many neurons are nearby, types of neurons, whether excitatory, inhibitory, also the types meaning which types of neurotransmitters they use, because different types of neurons use different types of neurotransmitters. The density of synapses, the density of axons and dendrites and the pairs, and the reuptake of neurotransmitters and neurochemicals, meaning when they're reabsorbed by all of the neighboring cells that are nearby. So all of this has to do just with the proximity, right? And each of these are statistical, right? They behave in a it depends sort of way. And then the topology. Topology is how the neurons are interconnected. Okay, and we've used simple diagrams here, but the real world brains are very, very complicated. We're talking about millions and oftentimes billions of connections. If you're talking about somebody speaking, speech requires probably the entire brain. So we're talking about tens of billions of neurons. So the interconnections become complicated, and the statistical nature becomes very, very complex. And by statistical nature, I mean the pattern of how the information flows from one neuron to the other, into this whole path over here, into this whole path, which splits up into these 4 other paths, and which of those 4 paths will it take, etc. All of that and the interconnections is based on the topology. and then the level of branching. And sorry, there's a typo here. This had to do with the extent of signal transfer. So, for example, whether it actually takes one of the branches or all of the branches, and so also the brain regions. Okay, that's another area. So, do they go to other brain regions? So, this is where the corpus callosum somebody asked about, but not just the corpus callosum. Even within a hemisphere, we have multiple brain regions. For example, our visual system is in the back. It can go to our frontal cortex, which is where we have a lot of conscious awareness and a lot of more sophisticated thought, executive function. So when we become aware of that thing that was moving in our field of view, that's not a snake, it's a tennis ball, the awareness would be in our prefrontal cortex, where we recognize it as a tennis ball. And to some extent, we also would recognize it in our visual system and in the parietal system, because the parietal system encodes space. So the fact that that tennis ball is moving through space, our parietal system is literally the neurons are tracking. It's moving right now. It's on the right side. Then it's moving center. That's center. That's moving toward the left. Our parietal system would track. So different neurons in our parietal system would information would flow from this cluster of neurons to this cluster of neurons to another cluster of neurons as the tennis ball moves in front of us. So that activity would happen. And then the specialty of those regions also would get affected. And then memory. This is another complicated area. So we're not going to go into these details here. This is kind of the end of the talk tonight. But the signal may excite at least latent memory that it will travel to. So, for example, this is a reason why sometimes when some signal happens, you remember something. You might remember a childhood thing that happened, or you might remember, it might be intentional that you want to remember. Somebody asks you, where did you go last night? You want to remember. So it may excite a latent memory intentionally or accidentally. Also, the memory with repetition, it may form or it may strengthen new memories. Again, we talked about the Hebb's maxim that when they fire together, they wire together. So this is a form of memory retention or repetition, memory storage. It may cascade to other regions. We talked about that earlier. That a lot of times a memory when we have a memory occur, what that basically means, what it appears to be, is that a large cluster of neurons, and these cluster of neurons don't have to be all geographically in the same location. The cluster can be this diverse cluster where you have 1 million neurons over here, you have 40,000 neurons over here, you have 1 billion neurons over here. All three of these clusters together, right? All three of these different size clusters together, they encode the thought of a parent, okay? And if you add in this and this other cluster, that encodes your mom. And if you encode these other clusters over here, they encode a parent, right? And if you encode different clusters, it might encode a grandparent. So for example, these three clusters together encode the concept of a parental figure. And then you can have specific differentiation of a mom parent or dad parent or grandparent, based on which areas of your brain activate together. And that's what memory, in a sense, is, that when we remember a specific thing, different parts of our brain are being activated and turned on. The active firing, when we saw that. And not just that they fire and they create this pattern, because memory, when it stays in our short-term memory, there's actually, I didn't go into this tonight, but there are actually loops in our brains, circuit loops. And these circuit loops are what allows us to remember things. And for example, short-term memory relies on a lot of circuit loops, because we can remember roughly 7 things. Most people can remember 7 things. You may have heard of that for a few seconds, about 30 seconds. And that's the reason why, because we have these circuit loops. So anyway, so also they can cascade to other regions. So when you have a memory activated, the memory of a parent, right, that memory can then go into other areas. And because you remember a parental figure or moms, let's say, that can activate something else. That, I have to call mom, it's her birthday, right? That may cascade and remind you of something else. One memory can remind you of another memory. Or it may result in daydreaming. So very complex types of behavior. So anyway, so that's it for the discussion. We'll try to wrap up now and have some questions and answers to those who want to, understand, or if you have your own comments. Joe, you're muted.
Yeah, sorry about that. Sure. Thank you, Sanjay. Now this will be time for any final questions or comments. I do want to get back to Dwayne's question from a little bit earlier. Does early life experience influence the pruning process? Can early experience alter opportunities for future learning? Go ahead.
All right. Yes. So early life. So pruning, and not just pruning. I mean, actually, the aspects of brain growth in infancy is a very, very complicated thing. So somebody earlier on made some comments which some of the things were not completely correct. You know, they said that when we're an infant, we really don't have any memories. That's not true. We do have memories. Some of those memories are encoded biologically. For example, the fact that we can suckle, we have a suckling instinct, or that our body knows how to thermoregulate our temperature, right? Infants can regulate their body temperature from birth. And they actually feel cold because in the womb they're very warm. All of a sudden they come out and all of a sudden the temperature has dropped. It's maybe 80 degrees in the hospital room, and literally it's cold for them. So and they start crying. So they feel temperature. They can regulate their temperature. So there are things that babies know from birth. It's not that they have no memories from birth. But what the person meant is they don't have memories of the real world, of the complex world. Although even that they do have. For example, they do have memories of their mother, what their mother smells like, because they've been smelling their mother in utero. There are chemicals, all kinds of chemicals all over the place all the time. So there are many types of training and imprinting that happens in infants. So as far as these early experiences affecting pruning process. So for example, the fact that when we're an infant, we can smell our mother because biologically we're literally connected to her, right? And we smell her, and that's actually a very important part of the growth process, because when we need to feed, we recognize her, we trust her implicitly because of that familiarity. Also the sound, we learn to recognize the sound of her voice because in utero we can hear a little bit of it, it's muffled. We also may hear the sound of our father if father is there, or any other adult, or other siblings for that matter. The child learns some of these patterns in utero, and when it's born, it will remember that. But for example, the sense of smell of the mother during pruning, that gets taken away because that's not necessary as much. So that's an example where early life experience, it's very important for the infant to have that ability to smell and recognize the mother through that, because initially the eyes haven't fully formed the ability to focus. The visual system isn't fully there yet. The first 2 months, it does come online, but for the first few months, the baby really can't see very clearly. It sees very rough. So it relies more on sound and smell. So the pruning process helps with that. As far as early pruning and early experiences helping future opportunities for learning, definitely. So one example is language, right? So an infant, by the time it's born, it starts to hear sounds, which it doesn't know what it is. It doesn't know their language, but its brain starts to make sense. Its brain starts to build, and all of the sounds that come into our brain, and I didn't go into this tonight, but I've talked about this in prior talks, that are the sounds that go into our brain. Actually, there's a region of our brain in the temporal lobe where the signals from our ears go into first. They actually, acts almost like a frequency meter, like you might have seen in some audio electronic equipment. Like it has this bar graph which shows the different low frequencies, middle frequency, high frequency. It can show. Our brain literally has something like that on both sides, both hemispheres. So each ear, and the signals that we hear actually gets broken up into different frequencies, and it gets passed on throughout the ear. So this is one of the reasons why our listening is so sophisticated, because our brain has from birth this ability. And so this infant is hearing all of these sounds. So it's starting to build patterns of the sounds of the language that are here. So if they're born into an English-speaking family, the English language has specific sounds that it has, and it doesn't have other sounds that other languages does. It's missing certain sounds. So the baby's brain is starting to develop and not record, but it's starting to build and grow in those specific sounds. And those sounds will remain forever because they're always going to be, as long as they're hearing, as long as they're being activated. So that's one example. But let's say that initially they grow up in this English-speaking area, and then after, in their teenage years, they move out of the U.S. and they go to another country where English is rarely spoken. And then they start to forget English. I gave this example earlier. Well, that early life experience, those memories are there, but the new language that they have to learn later on, it will start to crowd out, and it may overtake. It won't completely eliminate. It will crowd out and kind of make cloudy the original knowledge that they have of English. But what you'll notice, what happens in people who are like that, is that when they go back and hear English for a day or two, a lot of it comes back very quickly. So that's an example of that.
Thank you for that, Sanjay. I want to make sure we get to everyone. And again, type exclamation point in the chat for just even if you have questions or even your takeaway from this evening's meetup. I have Vanessa next.
This was a really good presentation, but my question is, let's say it's basically meaningless memory or event, but if there's something major significant, let's say you hear a bolt of lightning, you get a phone call saying a relative died, something significant, is it more likely to solidify that memory versus something menial that maybe you repeat, but like a jingle or something, it's not significant in your life? So can a major event like stimuli affect how a memory is encoded, or if it even is?
Yeah, definitely. What I didn't go into is the so there's a lot of depth that we can go into, but obviously I can't. Already we're running a little bit late, and I don't want to take up more time. But basically, the intensity of the signal that is what we're talking about. So, for example, if somebody dies, a close person dies. So let's take two examples. Somebody who's not very close. You hear that a famous actor or actress died, right? That probably is not somebody who's close to you, but you know them, you know of them. Or alternatively, somebody who a very good friend or a family member dies, right? So the person who's very close to you, when first time that you become aware of the idea that that person died. So what that means is that your brain would have to have the idea. So again, these clusters of different parts of your brain activate. So let's say it's a cousin, right, that died. And so the cluster of specific cells that activate together, that identify that specific cousin, they're activated. Right? And then there's another cluster that encodes the idea, the concept of death. Right? And when these things happen together, and that will happen because you're hearing, somebody's telling you, you're on the phone maybe, and you're hearing somebody tell you, oh, your cousin died. So your brain has decoded the audio speech, and it's activated those specific clusters of cousin, that cousin's name. It usually wouldn't be cousin, it'll be the name of that cousin. So it encodes the name of that cousin and the concept of death. And when both of those together activate, your mind will have the realization that that cousin has died. And then that realization, because these are all moving energy, moving information, and they're going all in different places in our brain, they will eventually also go into our emotional areas. And because those emotions will be very strong, because death, this concept of death is a very strong, it generates a lot of energy, right? Because we're all afraid. This is what when we say subconsciously, we're all afraid of death. This is the idea of that. So in that example, the amount of energy that's encoded with the concept of death will be higher. And so the amount of energy that goes into our emotional areas of our brain will be stronger. So that's an example. And so those neurons in the emotional areas will have stronger impulses. They'll be for longer time periods. They may have those loops that I talked about. Those loops will remain active instead of just for 30 seconds. Those loops may be active for 2 weeks because people stay depressed for a long period of time if close to a family member died. Right? So those loops keep regenerating, reactivating themselves for weeks and maybe months. So these are the complex things that can happen depending on the intensity and the types of signals that go into which areas of the brain. So for major events, yeah, definitely that can happen. And alternatively, if it's just somebody who's not that close to you, even though the concept of death is a strong emotion, because that person is not known to you or is not very close to you, there probably will be inhibitory signals that also go into your emotional areas that inhibit that strong feeling. And so you won't have a strong reaction to that person. You will have a reaction because death affects all of us, but it won't be that strong.
Thank you very much, Sanjay. We have Ella next.
Yes, along those lines, I have another question. If you have, if you are either on your own or in a therapeutic session or with any other individual, repeatedly going over something that was unpleasant, disturbing, that perhaps happened a long time ago, are you not strengthening those neurons? I mean, because the idea is that you're releasing, by going back to the event, you're releasing the trauma or whatever the case may be. But is that really the case? Because if you keep talking about it, are you not strengthening those memories and those neurons? And they're becoming more a part of your life.
Yeah, so that's a very good question. So The answer is a little bit complicated. It depends on several things. It depends on the therapist. So you described several situations. It could be a person, it could be a therapist, depending on who that person is. If they're a trained therapist, let's say they're a counselor who they're trained in trauma recovery or trauma counseling, that person hopefully understands that that can happen, that probably will happen. So they will probably use it very judicially. They'll be using it sparingly because they know that there's a possibility they may be strengthening the emotions and feelings around it. But also, there's so there may be purposes why they need to do it, because you one of the things that can happen, one of the things that in recovery, trauma recovery, that's kind of what the question is focusing on, is that we need to. So when there's trauma, what that trauma has done is it's encoded the automatic generation of strong signals in the brain. So for example, a certain music that you hear, okay, a certain song that you hear might be traumatic. So whenever you hear that music, it might remind you of an event or a situation or a person or whatever, right? So it could be anything for a given person. So in theory, you can't stop that music from existing in the world. You know, when you're walking on, when you're on let's say the subway or you know, or you're walking on a sidewalk or something, you might hear that song. So the therapist is going to want to reduce the effect of it on you. So one of the ways is what's known as exposure therapy. But exposure therapy has to be done in a very careful way, in that they have to keep most of you calm, and they have to be very cautious that they're monitoring to make sure that you don't become active or hyperactive or agitated or, you know, even hyperventilating or, you know, traumatized, re-traumatized. They have to keep, and this is the reason why in these types of sessions they're asking you regularly, how do you feel? Or like you're supposed to let them know how you feel. And they're you know, so you're right, it can be activating. There's another second part of this which I want to go into, is that there's a drug which basically, you know, people who know ecstasy, the chemical form is called MDMA. This is actually a very powerful drug, and it's being used in research settings now for exactly this. And what it does is it allows trauma and recovery therapy to be done, because what it does is that when the person is under this, and it has to be done by a psychiatrist, not just a normal therapist. It has to be someone who's medically trained. They give the drug to the patient, and that drug basically, it does certain things in the brain, that it reduces the intensity and it reduces the types of memories that, not memories, but it reduces the extent and the proliferation of those memories. It almost like it mellows out and calms out and slows down some of those automatic responses that a person has, and replaces them with almost positive emotions and feelings. And so when that's done, but it can actually replace them with negative. So this is why it has to be done therapeutically with trained people. You can't just take it on your own, because it actually backfire and make things worse. But this is a new type of experimental therapy, which exactly uses this aspect that you described, but it uses pharmacological agents to control the automatic response that a traumatized brain has, and lets the person feel relaxed. Because what the therapist has to do is they have to let you experience the traumatic, you know, input, whatever traumatizes you. You have to feel that. You have to sense it. You have to know it's there, but you have to not feel bad about it. You have to not feel the negative aspect, and that's where it has to be done correctly. They have to take away in the right way the negative side of it. So yeah, that was a very interesting question.
So I don't see any other questions. Do you have any closing thoughts that you would like to share? I mean, I think that this has been a fantastic presentation, a very, very detailed presentation as always. You know, one of the things is that maybe I would like just to ask, you know, how does our lifestyle impact how our brain actually works? And what are some of the things that we can possibly do to improve our brain health? You know, you talked a lot about how behaviors do impact how our brain behaves. Our behavior impacts how our brain functions. Just out of curiosity, you talked a little bit about learning a new language or something like that. What are some other things or anything that you could recommend?
So, I mean, this can be a very broad question. I'm going to try to restrict it a little bit, but
cool.
Brain health is obviously very important. It directly ties into our body's health, because our body if people who understand psychosomatic or people who understand the placebo effect, what it really means. What it means is that the brain literally can control and literally drive a lot of most everything in our body. And if you know that, for example, yogis, people who have been meditating for 10 years, they actually have learned to control their breathing, control their heart rate. They can change their heart rate by thinking about it. So the brain has immense control over our body also. So our health is tied to this. How do we make sure we're healthy? So the physiological side of health, nutrients, have a good diet, have good rest. Look at your brain functioning. Talk to people and look at what they tell you about you. Right. This is another aspect, because oftentimes, if we have something happening with our brain, usually we can't tell by ourselves. We have to rely on other people to tell us what's happening. And that's why the people that you are around, if you trust them, learn to trust them, and if you trust them, ask them questions. I mean, if you, depending on the type of relationship, obviously, but if they're close to you, then you may be able to ask them and let you know. If you see anything, if I do something weird, let me know, right? And
yeah,
it can be in a comical way.
I was thinking of even just like, experimenting with new things, in the terms of like learning to play a new instrument. Some of these things actually functionally change our brains, right? As they, so that impacts some of the neuroplasticity, the chemical neurons that we were talking about a little bit earlier. So I was trying to put it together in a more practical sense in some of those issues, some of those things that we can do. So yeah, you had mentioned even some of the physical exercise, the mindfulness parts of this as well, and how that impacts our brain. So I think that these are all really, really useful practical tools in addition to learning the science behind it. So thank you. Do you have any other closing comments?
No, I mean, if anybody has questions, we do this every month. And there are many videos that you're welcome to watch if you have specifics. I've covered a lot of things that most of the questions that people asked tonight. I've covered pretty much most of those in prior talks, and the talks are on specific topics.
And Ron, I did look for one of the presentations that Sanjay had given. I couldn't find it. It was I believe it was the one with the split brain.
It was, well, one was on the split brain. That was more than a year ago. That was almost a year and a half ago. And there were two that I did in vision. One of them was more on the artistic side of vision. The other was more on the basic biology of vision. Both of those were also a little more than a year ago, close to a year ago to about a year ago. I don't have the exact dates.
If I find them, I will send them to you through Meetup. I have, I could actually put the the neuroscience list in the chat here, while I'm also simultaneously announcing upcoming events. So let me just get first to upcoming events. Sorry, I have quite a few things going on here this evening. Well, thank you, Sanjay. Actually, just before, it was a fantastic presentation as always. It was really, it was interesting, and very much appreciate the time. Tomorrow night, we will be covering the Bhagavad Gita, chapter 18. It's the final chapter. I encourage everybody to attend. It's been a real pleasure having Yogeshwar, who calls in from India to help articulate what the meaning behind the verses in the Bhagavad Gita. That coupled with Srikant has been, it really has been truly an amazing experience. And that's both Thursday and Friday. And the next event after that would be writing collaborative poetry, and that'll be Monday night with Maritza, and followed by the Analects on Tuesday. And next Wednesday, I have to see, I believe it's Mike Amore will be presenting, but I don't know for certain. So what do you have in store for us next month, Sanjay?
So there's one thing that I need to follow up on. The talk we did last month, I have to do a part 2 on that. So I'm still not sure if I'm going to do that next month or the month after. But I want to do it probably before the end of the year. But that's where we're going to continue. Last month what we talked about was we were looking at, again, I mentioned we look at sometimes neural networks, computing type of neural networks. And we looked at the developments in the computing field, although it shouldn't be called computing. It's really the deep learning, machine learning side, and specifically neural network-based learning side of it. We looked at, for example, GPT-3, now GPT-4 will be coming out. We looked at a system called DALI. DALI 2 had come out earlier this year. And systems like that, which are basically starting to be able to do specific things that humans are very good at doing. So, for example, drawing. Humans are pretty good at drawing all types: painting, sketch figures, chalk, watercolors, any and every type of drawing. And the DALI 2 system, and there are several systems like that, have come almost as close as professional artists. And some professional artists actually say that these systems are better than them. So that's an example we looked at. Also, there are systems that do speech processing. And those systems can have natural conversations with us. And it's very difficult to tell if we're talking with another person or if we're talking with a computer, right? But these systems are very sophisticated. And so these systems we use to try to look at the models that they use and how those models actually map in with the models of the human brain and the overlap. And that helps us to explain and understand a lot more around what, with biological computational systems, biological brains, and the similarity with artificial brains. So there is a lot of strong similarity, and the systems are tied together. They are behaving in similar ways. Obviously, the artificial neural networks are a simplified version of biological neural networks. Therefore, they will show a lot of the characteristics, but it's uncanny the level of characteristics they're showing. And as the number of neurons grow, they're gaining more and more functions and capability closer to sophisticated humans. Right now, they're simple in limited domains. For example, only drawing and artistry, or only speech, only English speech, for example, things like that. But some of the newer systems that have come out in the last 2 months have started to meld these together. So the next talk I give on that will go a little more deeper and more sophisticated into that.
Thank you. So that's exciting. So I'm looking forward to that for next month already. Again, thank you. And thank you everyone for this evening. And everybody else have a great evening and we'll see you hopefully later this week.
Thank you, Joe. Thanks for your
Thank you.
wonderful questions, everyone. Yeah. Thanks.
Have a
good night.